PhD from Bangalore in 2003 and Masters as well as B.Pharm from Government College of Pharmacy, Bangalore. He has been teaching, as I already told you, from Goa in Goa College of Pharmacy, Panji. His research interests are impurity profiling, analytical method validation, and drug stability studies. He has many awards and fellowships to his credit. He has got Certificate of Merit, PD SAT Award for paper published in Pharmaceutical Analysis section of the Pharma Reviews. He has got St. John's Pharmacy College Award 2006 for best paper published under the section of Pharmaceutical Analysis in Indian Journal of Pharmaceutical Education and Research. He is a gold medalist of B.Pharm from Bangalore University in 1989. He has guided number of research projects at B.Pharm, M.Pharm and PhD level. His recent publications in last 7 years, he has not given for all the publications, but in last 7 years, he has 22 publications to his credit in international as well as national journals with high impact factor. He has published a book chapter. He has published 3 chapters in a book, Clinical Studies, Pharmacokinetics, Biomedical Analysis, and Bioequivalence. He is a sought after presenter and has presented in various conferences. He has also delivered lectures in quality improvement programs, continuing education program by different organizations, arranged by different organizations. In addition, he is called as a speaker in institutes in and around Bangalore. I'm seeing Alamin College of Pharmacy, Sri Siddhganga College of Pharmacy, and so on. He has coordinated UGC, DST, and AICTE projects and completed them successfully. The total grants sanctioned to him are about 50 lakh rupees. The theme of the project, which he has coordinated in industry-academia interaction, is guest faculty for BITS Pilani Master of Science contact programs for employees of Strides Arcola. The theme was cleaning validation of multi-purpose liquid oral facility. And there are further. Sir's biodata is so big that I'll not be able to finish it. So I request Sir to start with his presentation. Dr. Sanjay Paipi, please.
Thank you, Dr. Amrita Parle, Madam. It was nice that you cut short my biodata. And unfortunately, I should have edited and handed it over to you.
Yeah, short series are always better, sir.
It was my mistake. Anyway, it's nice that you gave a good introduction to the speaker, so that it gave me some time to really align myself in a direction where I can perhaps throw light on the topic that was required to be presented to the learners in the QIP program today. And I have taken this particular topic to give a background wherein this is something which is a hot cake of the day today in the industry. And there are many projects which are being honored and the works are getting published in this particular area in very good journals these days. I'm talking of those journals which are carrying very high impact factor. So what I'll do is in the next 45 to 50 minutes or so, I'm going to make a presentation on an interesting topic in the sense, uh, a topic, as I said, which is getting sold like a hot cake in the industry today. A lot of demand is coming these days with regard to new entities, especially entities which are considered as degradants from the pharmaceutical or from the drug substances and drug products. And there's a lot of, a lot of interest that has been developed, especially in the global arena when it comes to, uh, buying these kind of impurities for various purposes. As I go along, perhaps the listeners, uh, the participants of the program will get a fair idea about how many of these drugs, which are used as life-saving drugs, are actually leaching out certain compounds from within themselves because of the improbable storing conditions in which they are stored, or for some reason beyond the imagination of the formulator. So let me proceed with the presentation. I have covered this aspect from the point of the research interest that is shown both by academia and industry in this line. So I have titled it as Need to Regulate Degradation Product in Pharmaceuticals and In Silico Prediction of Toxicity for the Degradants. Now this is to fit the presentation with the objective of this QIP program. Uh, so let me proceed further. Let me just introduce the audience. Let me just introduce the participants to what degradants I'm talking of. Take a look at this paper. Now, if you look at this paper, which is published in a renowned journal, Chromatographia, it speaks about identification of degradation products and process impurities from terbutaline sulfate, a very old drug, terbutaline sulfate, by UPHPLC QTOF MSMS and in silico toxicity prediction. So if you look at this title, it becomes clear that when you say impurities, you are not just referring to impurities which are process related impurities, but those impurities which are formed from products as degradation compounds. And you see, to analyze this kind of mixture, I'll call it as mixture now. It's not just terbutaline alone. Terbutaline is accompanied by a number of degradants and process impurities. You cannot afford to use a very common instrument which may be there in majority of the laboratories spread in the spread across. I'm talking of the academic institutions, the laboratories of the academic institutions. You are thinking of highly automated devices. I'm talking of mass spectrometers. And you see, the objective of this particular paper, this particular study is you identify the degradation products and process impurities after their separation using a HPLC, and then you characterize them, and then you understand what it is, what is the structure of these compounds, and then try and predict if at all they are toxic to the person in case terbutaline sulfate has been administered along with these impurities and it has been consumed by an unsuspecting patient. You see, you subject the degradants, you subject the structures, identify through in silico methods whether these compounds are toxic. That is what it sounds like when you here when you read the title of the paper. I'll just go through the abstract so that those who are in research will get a faint idea about what this particular paper actually describes about. It's a rapid ultra HPLC method coupled with a hybrid triple quadruple time of flight mass spectrometry. Uh, uh, this is a method which will help in separation of degradant products and the process impurities of terbutaline sulfate. So what was done was this terbutaline sulfate, well, uh, as terbutaline sulfate, which is coming as a formulation, and with if due precautions are taken with regard to maintaining its stability, then perhaps terbutaline and only terbutaline may be present in the formulation. But in case if this terbutaline has not been handled well, either during the manufacture of the formulation or when it is stored in the shelf, then there is a possibility that this terbutaline, if it contains some susceptible groups, then it may undergo hydrolysis and form a number of degradants. So to understand that, this terbutaline sulfate may have been subjected to stress degradation through hydrolysis, by oxidation, higher temperature, application of humidity, application of heat, and all possible degradants which are formed when the molecule is subjected to these stressful conditions. Now they get into the product, and then you need to separate them. And when you separate them using a suitable mobile phase, you will be surprised to hear that they have been able to get almost 17 compounds. There were 17 other compounds other than terbutaline in that molecule, which will include which included 14 novel degradation products. When you say novel, perhaps they were the ones which were found for the first time to be present along with terbutaline. And this terbutaline sulfate was found to be highly susceptible to oxidative alkaline. This is their observation. And you see that when terbutaline is not stored properly, it's susceptible to cleavage and a number of products are formed. Now it's not the end of the study. The study now is when those compounds are formed, those compounds also must be chemicals, and they also must be having their independent activities, which can be either desirable activities or they can be undesirable activity. Fine, if they are desirable activity, if those activities enrich the activity of terbutaline, it is fine. But if there are undesirable activities attached to the undesirable, unpredictable activity, which is detrimental to the life of the patient, then it becomes an issue. So you need to predict the toxicity and perhaps what undesirable effects are done by these degradants. For that purpose, you need to subject these, you need to first isolate these components into, you need to separate them, and then you will have to individually inject them into suitable animal. You need a proper model for that. Well, it's a long study, but then who has the time to wait until, the product's undesirable effects are investigated? So today, just like tools are available, in silico tools are available for identifying newer molecules which are likely to possess bioactivity, in the same way, because these degradants also are chemicals, it is possible to predict the toxic effects, which again is an undesirable effect, which can be predicted with the help of in silico softwares. You have got one in this particular paper. They have used TopCat, which is a software which is very well accepted by many of these researchers, and it enjoys quite a good amount of support with regard to its ability to predict the different toxicities. Now, what they have done is basically terbutaline has been subjected to forced degradation, and the terbutaline samples have been kept for long duration of time. And then whatever degradants are formed, they have been separated by UHPLC, MS. And then once you get an idea of what these degradants are, you have subjected them to a study using an in silico system and predict the toxicity. This is what these fellows have done. This is what these people have done. You may find many researchers these days Going back to old drugs, terbutaline is not a new drug, but you see the year in which the publication has been done. It's in 2017. Now, people have found that even older drugs, many of the older drugs for which people never knew that they can coexist along with certain degradants and then reach the system of the patient and then start its own activity. People never expected that. And today, with development of automation, today it's possible to analyze components even if they are present in picogram, femtogram levels. I'm not talking of microgram level nor nanogram level. I am going to picogram and the femtogram levels. It is possible to analyze even if you have got certain components which are present in such lower concentrations. And it is possible today to locate substances present in such minute quantities. And that is the reason the automation which has developed, the technology which has developed these days, it is helping in looking at something which we had never looked before. And now we are able to understand why some of the what we assumed as side effects of a drug were perhaps due to the side effects were induced by certain degradants which were coexisting along with the main drug. Who would have imagined that terbutaline can coexist with 17 different degradants? You must have come across another drug, ranitidine, recently, which was in the public domain. And ranitidine, if you know, exists from decades. And, you know, it is something, that could be found only now in the recent past. And it's only because of the technology that we have today, we are able to see some things now which we never had the opportunity to see before. Who wants to see Mona Lisa, portrait of Mona Lisa in this form? You see, Mona Lisa portrait totally coexisting along with undesirable dots and aberrations. And if you remove them, you see, this is the one which has spoiled the portrait of Mona Lisa. If you adopt means to remove them, you end up getting the typical portrait of Mona Lisa. We adore that. Okay, coming back to the topic as such, I'm looking at the impurities which are listed here. This is one diagram which shows types of impurities, the different types of impurities that you can predict or you can expect to be present in the formulation. Whenever you have something else other than the drug in the formulation or in the API, you call it as an impurity. It can be organic, it can be a solvent which has remained, which was used during the formulation or which was used during the making of the API, but it still continues to remain. Or it can be something which is inorganic in nature and is responsible, and it was used for some reason when the formulation was being made, but then has remained in the formulation because due care has not been taken for removal of these impurities. Now I'm concentrating only on that impurity which you see right at the top end. These are the degradation related impurities. Well, when you say impurity, impurity can be organic. When it comes to organic, it can be the degradation related impurity, or it can be process related impurity. When you say process related impurity, these are generally the predicted impurities. We always identify them as the predicted impurities because we know what is the source of these impurities in the drug substance. It can be a starting material. I'm talking of an API here, something which has been used in the making of the API, or you know that these APIs come from several steps. It can be 3, 4, 5 steps, and some intermediates can continue to remain till the final product is reached. Then also you call it as a process related impurity. It can be a byproduct or it can be a side product which continues to remain along with the main drug. All these are process related impurities. Their presence, their formation can be predicted if you are a good chemist. But I'm not concentrating on that part. I'm concentrating on these impurities which are formed in the API because of some degradation. You know, an API is a chemical and it has got several sensitive or susceptible units in it, which can be tapped by an external medium, and these portions, right, they can just give up somewhere here and there and can result in a compound which is not the drug. So you call it as an impurity. So these can be degradants, you call them as degradants, not the process related impurities. These DRIs, what I mean here now, they are the ones which are formed due to degradation of the API. Then you call it as the API degradants. It is formed since there was some susceptible group which was reactive in presence of maybe moisture, in presence of oxidants, in the presence of heat. The higher temperature would have resulted in some kind of damage to the molecule, which would have resulted in the formation of degradants. So under environmental conditions, there could be formation of several degradants. There may be, when the formulation is prepared or manufactured, there can be some interaction between the excipient and the drug itself, and this interaction could have led to formation of a degradant. That's also a degradant. There may be certain residues which are present in the API, and their interaction could have resulted in the formation of some other entity, or can be an interaction with the container, and that can result in the formation of a degradant. And these degradants, which are very difficult to predict, need to be analyzed in order to find them and then characterize their structures. So what's an impurity? If I have to go one step back, since I've been repeatedly using the term impurity, these are any components of a drug substance that is not the chemical entity defined as a drug substance. And in addition, if it is a drug product, it can be any component that's not the formulation ingredient. So when it's not a drug substance, when it comes to API, when some particles in the API are not the ones which they are supposed to be present in it as a drug, then you call it as an impurity. And even in the drug product, in the formulation, if it is something else other than the known excipient along with the API, then also you call it as the impurity. So we can classify these impurities as the process related impurities, the degradation related impurities, and the interaction related impurities. I'm taking the last two in this. I'm concentrating on the degradation related impurities and the interaction related impurities here. You know that impurities, if they are present, they are the ones which will affect the safety and efficacy of the product. So you can as well consider that they are the critical quality attributes, and if in case if they are present in the drug substance or the drug product, they have the potential to affect the safety as well as the efficacy of the product. Now, as I said, degradation products, how they are formed, why they will appear along with API. Well, it can be an interaction with the environmental conditions, which can be either light, it can be temperature, or it can be moisture, the humidity, all these environmental effects to which the drug is going to be exposed to can result in some kind of degradation, which can happen within the formulation if it is not stored properly and can lead to formation of certain degradant products. There's another possibility of these degradant products which can be formed when the formulation of the API, when that is getting manufactured, if due precautions are not taken, like certain drugs are sensitive to heat or they can be sensitive to the moisture. In such cases, it's possible that unpredictably there can be the formation of some kind of impurity which will continue to remain and then is likely to be administered to the patient. And when the patient consumes the drug which unsuspectedly is containing that impurity, the impurity is also a chemical, and maybe it can elicit some kind of undesirable response. As long as it is desirable response, or if it if it's totally inert, then there is no issue. But if it elicits some kind of undesirable activity, then definitely care has to be taken to ensure that such degradants are not present in the drug formulations. The ICH today has norms through Q1 AR2 and Q3 BR2 to take care of these degradants which are formed after systematic stability studies, the stress stability studies which are conducted, in which drug is exposed to moisture under different conditions of acid, base, and the neutral conditions, the drug after exposure to light, higher temperature, and moisture. So we are getting into the area slowly, we're stepping into an area which which needs a due care to be taken, because you may end up with degradant. The API may have beneficial effect, but the degradant which is formed in the formulation, that might elicit some kind of toxic effects or teratogenic effects. There are thresholds that have been declared by many of the regulatory issues, and whenever these degradants are formed to extents beyond the threshold, then it is necessary that there is detailed investigation or analysis which is required to be done. I'll come back to this towards the end of the presentation. Now, what is the significance of this impurity study? Gone are the days when people used to only think about how much is the purity of this particular drug. Today, the monographs, if you look at the monographs, the current monographs, they will not only talk about the purity attributes, they also speak about the impurities and the related substances that can be present. Now, these related substances and the specified impurities which are listed in the pharmacopoeias, these are the ones which I am not talking of. I am talking of something different. The degradants which are formed, which will continue to remain along with the API, and then are administered into the system of the patient. The fundamental requirement for license of a pharmaceutical product today expects the drug to be present in the purest possible form. With even if there are degradants present, they should not elicit some kind of toxicological response. Well, when I say toxicological response, you should also know that when you say something is toxic, it can be genotoxic effect, or it can have some kind of mutagenic effect or the carcinogenic effect. Many a times it has been seen that when formulations are made, the excipients or the residual impurities, they cause instability of the API and the drug product. And many a times, the impurities originate because of the interaction of the excipients and certain residual impurities, which can be specified impurities, when they interact with the APIs. Many a times the functional groups in the APIs, they are susceptible to degradation on account of hydrolysis, oxidation, or some kind of polymerization effect. I'm just giving you a small list of popular drugs which can degrade and result in formation of certain degradant products. And you see in the last column, you can read the kind of toxic effects that these degradation products which are formed can initiate in the human system. Just to read a few, you see tetracycline, the most popular antibiotic, tetracycline, it can basically degrade and form the 4-epiandrotetracycline, and the isoniazid used for tuberculosis can contain hydrazine as its degradation product. Ascorbic acid can have furfural as a degradation product. Ibuprofen can have 1,4-isobutyl phenyl 1-ethanol as a degradation product. And you say lidocaine, an aesthetic, can contain 2,6-xylidine as a degradation product. Proguanil can have 4-chloroaniline as a degradation product. Rofecoxib can have a methyl sulfonyl phenylfuranedione as a degradation product. The presence of these degradation products along with the active drug is the focus of this lecture. You know, this degradation products, if they go undetected, you can imagine what kind of effects that these degradation products can elicit in such patients who unsuspectedly are administered with these drugs. I have put a few effects there, although I found that some of the effects are not very damaging to the patient, but the ones which I found damaging to the patient are the ones which are listed across in each of the rows. Here are some of the popular degradants which have drawn public attention during the last decade. One of them is 4-nitro-trifluoromethyl-aniline, which is formed from a non-steroidal anti-androgen flutamide. And one more degradant coming, which it has drawn attention of the public, is the presence of this 2,6-dichloroaniline. It's one of the starting material which goes into the making of diclofenac. And this diclofenac is supposed to even break down and form 2,6-dichloroaniline as an oxidative degradation product. So this aniline, which is an amine, can be carcinogenic. Basically, amines have been proved to be carcinogenic, can initiate the disease of cancer. Most recently, we all came across this NDMA, the N-nitroso-dimethylamine. Uh, When popular formulations of multinational companies, were banned, had to be withdrawn. Uh, this was a carcinogen, and you see, even though the finished product did not contain this NDMA, but certain environmental conditions like higher temperature was said to be the reason for a release of this NDMA due to the degradation of ranitidine within the strip packages, and that was one of the reason that that has led to a large scale withdrawal of this ranitidine formulations from the market. There's one more which not very recently, but some time ago, maybe a decade ago, which drew public attention was formation of this 1,2,6-dichlorophenyl indolene 2-one, which was formed whenever this injection of diclofenac sodium, if present in the alkaline medium, when it was sterilized, this diclofenac within the ampoule was giving rise to this compound, dichlorophenyl indolene 2-one, which was a degradant and which had undesirable side effects. So unsuspectedly, you see, these degradants can form within the formulation containers, even though they have left the manufacturing facility with a seal that they comply with all the regulations, purity regulations. But in the market, when they reach or during the final stages, maybe I'll call it as just before, getting administered into the patient, there can be formation of some degradant which can happen at the level of the retail pharmacist, or when it is in the hands of the customer or the consumer or the patient. And if at all there is formation of this kind of degradant, which is not a good sign when it comes to, not a healthy sign when it comes to, the toxic effects of the uh, formulation is concerned. You may be wondering what else can be the source for impurities in these formulations. It has already been proved in many cases that some other component which goes in the formulation as an excipient other than API can also be one of the reason for formation of the degradants, like the peroxides and the high water content in povidone, which is used as a which is used as a excipient. The peroxides and the high water content in povidone and polyethylene glycol can become a source for degradation of the API which is present in the formulation, or the antioxidants of the magnesium stearate, the aldehydes in lactose, benzaldehyde in benzyl alcohol, formaldehyde in starch, lignin and hemicelluloses in microcrystalline cellulose. Now, if the industry concentrates only on purity of APIs without giving due recognition to purity for the excipients that are used, then you are administering these triggers in the formulation for them to undergo degradation and release harmful substances. So as I said, the chemistry of degradation is basically, it can be oxidation or hydrolysis. These are the two major reasons where many of the drugs which are susceptible to oxidation reaction and hydrolysis can break down and form certain other components, and they coexist along with the API, and can show undesirable effects when administered in the patient. I'm not going to take much time here. Hydrolysis, we all know, it's a reaction where the water molecule which is present, it has an impact on bond cleavage. And, the esters, for example, if a drug contains an ester group, that can get broken down into an acid, or if an amide group is present in the drug, that can break down and form once again an acid. Well, esters are more susceptible than amides to undergo hydrolysis. I've just given an example here for you to appreciate the fact. You see that you have lidocaine and procaine. These are the two molecules which are susceptible to hydrolysis. When you think of these molecules, procaine contains an ester group. It's not commonly used these days because of this reason, the stability reason. It's a good local anesthetic, but because it is an ester, it is susceptible to hydrolysis, and the vital ester link is broken because of the hydrolysis. Even lidocaine, it contains an amide bond, and it is susceptible to hydrolysis. And it so happens that although it's an amide, it's less susceptible to ester, but it can still undergo hydrolysis if not properly maintained, and result in degradants of procaine and lidocaine, which are not active to the extent of the activity exhibited by procaine and lidocaine. You see the amide group here in see CONH, the amide group, as you can see in lidocaine, and the ester group, as you can see in procaine. And you will see that when these formulations hit the market, and if they are not protected properly, they break down into corresponding amine. The lidocaine breaks down into corresponding amine. Amines are once again carcinogenic in nature, and procaine breaks down into an acid, which has no activity, intended activity of procaine. So there are certain other functional groups, and if they are present in drugs, they get very easily degraded in the presence of moisture alone. Like for example, the imino group in diazepam, the acetal group in digoxin, the sulfates in heparin, the phosphate esters in the hydrocortisone sodium phosphate. The unfortunate part is when degradation takes place on account of presence of moisture in these you can say very popular drugs, it may so happen that the potency of these drugs either disappears completely or gets reduced remarkably. So the intended application is defeated, and we are actually administering a substandard drug without our knowledge. There are many compounds which are susceptible to degradation, and in one or the other way lead to formation of an amine. And whenever amine formation takes place, then it's quite risky. As you can see in this example, lidocaine can form, you see, 2,6-dimethyl-aniline, which is a carcinogenic. Similarly, prilocaine on decomposition can give rise to orthotolidine. So the regulators have understood the importance of these kind of reactions in drugs, and has the implication of that is that they expect that these impurities are likely to be present in such drugs which originate or which are susceptible to undergo hydrolysis. And then whenever the suspect is that there is formation of amine, lot of restrictions are imposed, especially on these kind of degradants. They are considered as either degradant, either as degradant impurities, or they can assume the role of a specified impurity provided if the starting material happens to be the same compound. Like if lidocaine is synthesized from 2,6-dimethyl-aniline in one of the synthetic routes, and it so happens that because of degradation, lidocaine gives 2,6-dimethyl-aniline. Now 2,6-dimethyl-aniline becomes a process-related impurity as well as a degradant, and it can find a place as a specified impurity in the pharmacopoeia. There are very strict regulations which have been, and you see the regulations today, if the regulatory bodies are trying to tighten the screws in such a manner, because it's possible to visualize the degradants on account of the automation that is available to us today. The kind of automation that is available to us, the liking of MS, for example, has thrown light on many invisible things that was never witnessed earlier. So qualification is one of the important parameter that the regulatory body expects any manufacturer or any any scientist who claims to have discovered something new to mitigate some kind of disorder. The qualification is basically a process of evaluating the safety data and establishing the acceptance criteria. Now I'm slowly moving towards evaluation of the toxic effects of the degradants which might be formed in the formulations or which may be present along with the API. Even the ICH M7R1, uh, it's a regulatory requirement for control of genotoxic impurities and degradation products, entitled as control of DNA reactive mutagenic impurities in pharmaceuticals to limit potential carcinogenic risk. Some of the chemicals that have been listed, which are likely to be formed because of degradation, you can see acrylonitrile, benzyl chloride, bis-chloromethyl ether, 1-chloro-4-nitrobenzene, paracrecidine, dimethyl carbamoyl chloride, ethyl chloride, glycidol, hydrazine, methyl chloride, parachloroaniline, dimethyl sulfate. So you see, these are the potential compounds which are likely to be present, which could be either a degradant or it can be a solvent which may have been used in the manufacture of that formulation. And it as an impurity in the formulation, if it is present, they are considered as the carcinogenic impurities. Many regulatory bodies, have now proposed many guidelines. In the last 15 years, you see there are more number of guidelines to cater to basically scrutinize the impurities that are present in drug substances, drug products, and the biological products. You can see in this particular table, it classifies the kind of regulations that are applicable, that are promulgated by ICH, FDA, USP, EMA, the European Medicine Agency, and you see that most of them have got stringent regulations when it comes to the detection and disclosure of toxic potential of the organic impurities, specifically the process related and the drug related. When I say drug related, I'm talking with regard to the degradants which may be present along with the drug. The residual solvents which continue to remain along with the formulation even after the formulation is ready for marketing, that also needs to be analyzed. And some of these solvents which basically continue to remain present along with the formulation, their limits are expected to be kept under control. The major issue basically which EMA, ICH, and FDA look for is basically the genotoxic impurities, which if proved that they are genotoxic, then there are very stringent regulations to minimize the presence of those degradants, to ensure that those drugs don't give up those degradants within the formulation, which eventually turn out to be genotoxic. The ICH recommends 5 major steps for the management of degradation products. If degradation product has been proved as an impurity in the formulation, then it is necessary that that particular degradant has to be characterized. It is necessary that you identify the way by which that particular degradant has found a place in the formulation. So it is necessary to evaluate the pathway and then disclose the likely way this particular degradant has found a place in the formulation. And then if the amount of these degradants present go beyond threshold, then it is necessary that it has to be reported, and then means have to be found in order to keep the impurity under control. Every regulatory body today is keen to target the impurities that are potential genotoxic, mutagenic, and carcinogenic. You see that it's not happened in the recent past. Way back in 2006, the EMA, in 2008, FDA, they have come up with regulations, they have come up with guidelines specifically targeting these genotoxic, mutagenic, and carcinogenic impurities. The genotoxic impurity, if it has to be proved, well, there are many experimentations, there are many classical laboratory exercises which are found in the literature. They are all very long. They are very long, and it becomes it becomes not very easy for a drug manufacturer to launch his drug just by when the regulatory body has guidelines which has to abide with the degradants which are likely to be genotoxic. The manufacturer in such case will have to resort to laboratory tests and then come up with certain disclosures whether the degradants that are likely to be present in a particular formulation can elicit a genotoxic response. So there are many experiments that have been proposed, laboratory exercises that have been proposed, but the biggest issue is the time duration. I've just listed out a few toxic tests that are advocated in the guideline, but it so happens that when you start working on these particular laboratory exercises, there is an absolute delay in the launch of the new products. So what is the solution for avoiding such long delays? The one way of overcoming that is to use the in silico techniques which have been permitted by the regulatory bodies and verify whether these degradants have the ability to elicit genotoxic effects, and then perhaps apply the new product, the new formulation for marketing with a disclosure that that contains degradant which is unlikely to cause genotoxic effects. Now, both the impurity study is required to be conducted to cater to both requirements. It's the regulatory requirement. Many industries indulge in impurity study, not just for supporting the regulatory requirement, but for their own internal purposes, wherein if they have to claim that their product is superior to the product which is manufactured by their competitor, then they disclose certain specifications which give a fair idea about the involvement of these kind of studies, so especially when it comes to marketing of those drugs which are likely to contain certain impurities which are likely to be detrimental from the point of genotoxicity, detrimental from the point of the mutagenicity, or for the purpose of genotoxicity. For the purpose of regulatory requirements, in order to prove the quality and safety of the products, to perform the method validation, to disclose the acceptance criteria determination, for the purpose of expiry date, retest date, and the shelf life evaluation, the impurity study from the point of regulatory requirement is required to be done for the purpose of ensuring stability, to determine the threshold limit evaluations, etc. From that point, impurity study is a must when it comes to the regulatory requirement. When a lot of noise was created when the nitrosamines were first found in ranitidine very recently, there were several questions which were asked to FDA Like one of the questions is the presence of nitrosamine in drug a new problem? Why have there been so many recent reports of drugs containing nitrosamines, etc.? There was a very interesting thought which was passed by FDA. I have put it so that the participants can get an idea of what FDA thinks today and in which direction the FDA expects the industry to comply with for their products to be passed. You see, FDA, to an answer of this question, said FDA has ongoing assessment, surveillance, compliance, and pharmaceutical quality efforts across every product area, and we will continue to work with drug manufacturers to ensure safe, effective, and high-quality drugs for the American public. When we identify new and previously unrecognized risks to safety and quality, we react swiftly to resolve the problem, as we have done in responding to recent findings of nitrosamine in certain medicines. This second paragraph is very critical. Today, we have better testing methods than ever before, and we know what to look for in products' chemical structures. You see, the uh, it's little harsh, but then it is the truth. The manufacturing process, and that may increase the risk of forming low levels of nitrosamines. Improved technology enables us to detect. See, they talk about the technology. What we could not see before, we are able to see it now. What we could not forecast before, we are able to forecast it now because of the improved technology. It enables us to detect even trace amounts of impurities in drug products, and maybe the reason why more products have been found to have low levels of nitrosamines. The agency has strict standards for safety and effectiveness and quality, and our staff makes every effort to help keep the US drug supply as safe as possible. We also work closely with international drug regulatory agencies so that we leverage resources and testing done outside the US, which can help inform testing of the US drug supply. As our investigations and testing continue, along with the investigations done by international drug regulatory agencies, we may find low levels of nitrosamines in additional drugs. The story doesn't stop with nitrosamines. What I have learned is they have found many more equally toxic compounds like nitrosamines, and they have started finding these toxic degradants, as I can put it in the right manner. They have started locating these degradants in many more age-old drugs which we are still using and which are still in the market today. Now, understanding the importance of the degradants which have the ability to elicit toxic response, it becomes necessary, it becomes imperative that a manufacturer or a researcher will have to provide data with regard to possible degradants that might form from the drug, along with the toxic potential that it possesses. Now, it so happens that if you have to go on doing, if you have to prove that your drug is effective and contains such degradants which are non-toxic, then animal study becomes important. And animal studies to carry out and to get results out of it, very long. It takes long duration. So better way today, just like, it's possible to predict drugs which have got good activity through QSAR modes. The molecular modeling and QSAR has come as a rescue for discovery of new drugs. In the same way, this in silico technology, toxicology as a technology has come as a rescue because it is possible to predict the toxic effect of these degradants with the help of the computational methods. So the toxicity assessment tool uses computational methods to analyze, simulate, visualize, and predict the toxicity of chemicals. The toxicity assessment tool it aims to complement the existing toxicity tests to predict toxicity, prioritize chemicals, guide toxicity tests, and minimize what's more important is this, the minimize the late stage failures in the drug design. The computational methods have a unique advantage of being able to estimate chemicals for toxicity even before they are synthesized. In the drug discovery, when you propose that this is likely to elicit action through molecular modeling, then before that molecule is made available in the wet laboratory by synthesis, it is possible to predict what are all the possible degradants that can be formed from that particular drug, which is likely to have bioactivity. And then you can even predict the toxicity. So this kind of toxicological study goes hand in hand with the new drug discovery when it comes to in silico, the contribution of the in silico systems. So you discover a new drug before you propose that, this is a drug of the decade or a drug of a century with regard to treatment of such and such disease. You can even exercise and predict what is likely what are the degradants that are formed out of the new proposed drug, and then you can even find what are likely to be the effects, the toxic effects that are likely to be caused by those degradants. So these computational methods have now come as a rescue, and you will be surprised. The kind of tools that you are using are similar to the tools what you are using for your new drug discovery. You have databases here, you have a software, you have simulation tools, you have modeling methods, you have the modeling tools, you have the expert systems, you have the visualization tools identical to what you are using for new drug development, new molecule development. The only difference is, or the similarity is, the new drug is a chemical, the degradant which is formed is also a chemical. So obviously, no surprises here. If the drug has beneficial action, the drug will have an undesirable action here too. And of course, when that undesirable action becomes a toxic effect, then perhaps eyebrows are raised. These are the tools. You see that they are almost identical to what you see when it comes to new drug discovery. You have databases, you have to identify descriptors, you have to go for simulation, you go for modeling methods, and you use the modeling software. You see, these are all the tools which you are using for the new drug discovery. And then visualization. You see, when you are going for prediction of beneficial activity, it is possible to predict toxic activity also. So if you can predict the most likely beneficial activity, conversely, if you disclose, then it is possible to even identify the undesirable effects. There are many tools, the sub tools which go into this computational studies. The goal of the in silico toxicity is to predict the chemical toxicity through computational modeling. Here I am using the word QSTR, I'm not using the word QSAR. Basically the quantitative structure toxicity relationships, and you write algorithms, you predict algorithms which can help you in generating the toxicity data. One of the earliest tools that was used was the structural alerts. Even the EMA and the US FDA guideline does mention about the use of these structural alerts. You see, they recommend the use of chemical structural assessment as a valid means to predict the toxicity potential of drug impurities, including degradants. The concept of this structural alert was first elucidated by Ashby and Tennant, who investigated the correlation between chemical structure and the DNA activity for about 300 compounds based on the Salmonella carcinogenicity assay. According to Ashby et al., the structural alerts are the structural features of a compound that can be correlated directly with the ability to elicit the carcinogenic response. Some of these structural alerts, basically these are the moieties which are considered to be the undesirable moieties. If they are present, they can elicit some kind of undesirable response. They can be amines, they can be carbonyl compounds. And you see that when you identify possible degradants in the degradant database, 70% of those structural alerts were either aldehydes or carbonyl compounds or the amines. I'm just giving you a few examples. I'll just take a few more minutes here. You see acetaminophen, paracetamol. One of its a structural alert, one of the degradant of acetaminophen, that is paracetamol, is paraminophenol. Paraminophenol itself goes into making of paracetamol. So it can become a process related impurity or it can become a degradant. Now you see This para-aminophenol has the amino group, right? And these amines, as you can see in this particular slide, the anilines or the amines, the aromatic amines, right? They have the potential to elicit carcinogenic activity. So if there is a para-amino, or you can say aromatic amino group present in the degradant, that becomes an alert. So there is a structural alert when it comes to one of the degradant of para-acetaminophenol, that is paracetamol or acetaminophenol in the form of para-aminophenol. Likewise, there is another one, chlorhexidine. It's an antiseptic antibacterial. You see, it can generate para-chloroaniline on account of hydrolysis when it is present in aqueous solution. And you see this now, chloroaniline. This again contains that aromatic amino group. And this degradant, which is formed from chlorhexidine, you have an alert here, a structural alert, which happens to be an amine. PCA, in fact, parachloroaniline, is quite toxic because it has already been experimentally shown to produce lesions in animals, especially kidneys, spleen, and liver. So whenever amine degradants are likely to form, the regulatory bodies have enforced stricter regulations, and their limits, as you can see in USP, the limit of PAP, which is the para-aminophenol, is about 0.005%. So anything that is found beyond this, the batch doesn't pass. Similarly, for parachloroaniline, the limit is 3 parts per million as per the USP. There are other toxicity predicting models which are used, like the rule-based models. Of course, these are not enjoying the privileged support of the regulatory bodies, because they appear a little vague in the sense of predicting the toxic behavior of the degradants. In this rule-based model, there are two types. One is the human-based rule. The other one is the induction-based rule. The induction-based rules propose a hypothesis about association between chemical structural properties and toxicity endpoints, which cannot be predicted by the human insights. Uh, well, the regulatory bodies have not found good rationals through many of the laboratory experiments which have been done in order to support this model. The next one is the chemical category. Like, you categorize the molecules as the one containing amino group, the hydroxy, the phenolic group. Then, you say that it's a carbonyl compound and so on. So based on chemical category, the degradant which contains the functional group can be identified, and then, its ability to induce the toxic effect can be predicted. The read across is another tool where you predict the unknown toxicity of a chemical when compared to a similar chemical, which is already known for toxicity. So if there are structural similarities in the degradant to already existing chemical or its analog, which has known toxicity, then we call that model as the read across. It's like extrapolation. If there are let us say two amino groups present in a particular degradant and is already proved to be toxic for its carcinogenic effect, and a new degradant which is formed which also contains two amino groups, based on that you can extrapolate from the previous search and claim that this particular molecule also is likely to show carcinogenic. There's a risk of carcinogenic effect to be shown by this particular degradant. So it's called read across, which again hasn't enjoyed much privilege of the regulatory authorities. The other one is the trend analysis. You just it's a method of predicting the toxicity of a chemical by analyzing the trends. We call it as the toxicity trends, like increase or decrease in the carbon chain length. To give you an example, if methyl chloride is likely to show some kind of effect, ethyl chloride also is expected to show the same effect, because the difference is only with regard to change in the carbon length. That's called as the trend analysis. That's another tool which is slowly or gradually enjoying some kind of attention when it comes to prediction of the toxicity. There are many more models like this. One is the dose response and the time response model, then the pharmacokinetic model and the pharmacodynamic models. The The pharmacokinetic models relate chemical concentration in tissues to time, estimate the amount of chemicals in different parts of the body, and quantify the ADME process. Well, this requires some degree of laboratory data, the experimental data. They are more reliable, but then they are again time consuming. They cannot be compared with the kind of data that you can generate out of, let us say, the QSTR, but it requires thorough investigation and requires some laboratory study in order to come out with data under these kind of models. The pharmacodynamic model relate to biological response to concentration of the chemical in the tissue. The toxicodynamic models are the pharmacodynamic models that relate toxicity to concentration of the chemical. The pharmacodynamic models that are based on anatomy, physiology, biochemistry, and biology are called the physiologically based pharmacodynamic models. I have to emphasize here that the pharmacokinetic and pharmacodynamic models, they are not the computational models as such, but they require extensive, these are the normal, the models other than the computational models, which will provide laboratory data in order to prove whether the degradant is toxic or to the extent of toxicity that can be exhibited by degradants. I have found a number of websites today which can the number of websites which can give you an idea with regard to the ability of the molecules to elicit pharmacological action. That is, I'll say, beneficial pharmacological action. In the same way, there are many websites today which can give you a fair idea about the undesirable activity or the non-beneficial function of the chemical compound as such. Now, degradant also is a chemical. So you identify which is the degradant based on the, based on the MS study, the LCMS study. Try to jot down the structure of it, and then you feed the structure appropriately, and it is possible to vaguely expect, in many cases, approximately expect, I'll put the right word, the kind of toxic effects that these molecules can exhibit. Here is a small list of such data sources. Uh, this is something like what you do in your computational studies when it comes to drug discovery. In the same way, this is what you do when it comes to computational study with regard to prediction of the chemical toxicity. Some of the popular ones are the ToxNet, the ADMET SAR, the ToxCast, the EcoTox, very commonly or very popularly heard if you are in this area of predicting chemical toxicities. Uh, the drug bank and all will provide you a fair idea about the possible toxicity effects, but they may not help you necessarily in predicting the toxicity of the new degradants if you have discovered something in your laboratory. I think I have just given a bird's eye view of the importance of the degradants, its formation in the drug, and if at all they are formed, then what guidelines, what are the likely guidelines that have been thrown apart by the regulatory bodies today, and how there has been a development in the area of computational studies in order to identify potential toxic effects of these degradants which are formed in the drug substances and the drug formulations. So I think I have just seeded a thought in my young researchers group in this QIP program. You can actually work on these kind of projects at length. This is a culmination of basically pharmaceutical chemistry, pharmacology, pharmaceutical analysis, and a little bit of medicinal chemistry. You see a culmination of thoughts from various branches of pharmaceutical chemistry, and that can lead to full length projects which can be taken up even for PhD studies. Uh, with that intention, I just wanted to give you a bird's eye view of how you can use these kind of projects, especially target at degradants. You can take age-old molecules, as many researchers are doing these days, and then identify, because nobody would have viewed the possible degradants in old molecules which are popular even today. You can take up those molecules, identify degradants. The only drawback here is that you should have access to the LCMS, and then develop suitable methods, identify the degradants, and then sketch them based on the chemistry knowledge, sketch them, put their structures, use this, the chemical toxicity programs in order to predict the toxicities. I think this is what I had to brief my generation of researchers when it comes to area of degradants and their potential toxic effects, which are likely to damage the potential of the active drug. Any questions?
Thank you very much, sir. Sir, you have done a very wonderful presentation. So now the session is open for the delegates for the question answers if they are having any queries.
Are there any queries with the participants? Dr. Pai would be happy to answer the queries. I think Dr. Sanjay ji, your presentation was so well structured, covering each aspect, so that all the points are clear in the minds of the delegates, and that is the reason they do not have any queries. But
ma'am, I have a query for sir.
Yes,
okay,
yes, please.
great. Please put your video on.
Yes. Good evening, sir.
Yeah, good evening.
Sir, recently I've completed my doctorate and my topic was force degradation studies only.
Okay.
But I'm mostly an analytical person.
Okay.
But I have a knowledge of mass degradation, not to the level which is required. But I've worked on 4 different APIs, which were recently launched by FDA, US FDA, in the past 5 years.
Okay.
And I have the degradation profiles with me. Okay. Now, when I listen to your presentation, it is like if we know the structure, we can go for thinking about the
prediction of toxicities. Prediction of toxicities.
Yes. Yeah. So then, how exactly to look into that aspect? Can you just throw some light on it, sir?
Yeah. Because if you look into these programs that are available, these are programs on lines with the programs that are available for your drug discovery. So what they do is, you'll have to identify those alerts in your molecules. Check whether those alerts are present. When I say alerts, these are the structural alerts which are present. There is already a pre-fed information in these programs. Okay, some of the programs wherein if there is presence of this particular group, right? So what they have done is known toxic compounds, data from known toxic compounds has already been fed into these programs. Yeah. And then if such structural similarities are present in your degradant, it is likely to exhibit this and this kind of toxic effects. It is shown like that. So what knowledge you require is to identify the structural alerts in your degradants. So you'll have to feed those molecules. There are programs which will identify structural alerts based on pre-fed information. If like a pharmacophore, what you talk in terms of drug, pharmacophore is something which is required to exhibit activity, positive activity. And negative activity also can be, your pharmacophore can be, the understanding of pharmacophore can be extrapolated even for a drug which can elicit a response, whether it is desirable or non-desirable. When it becomes non-desirable, it becomes toxicity, right? So you are feeding the information about your drug in such programs, and then these programs start correlating with the structures which are already available in its memory and tries to identify those alerts in your molecules and then it will show it is the presence of such and such entity in your molecule that it can elicit this kind of toxic response. It gives you a vague idea, a rough idea that yes, this structural alert can be a cause for a possible toxic effect in this particular degradant.
Yeah.
Okay, if that data is not available, then what you have to do is there is a possibility wherein you will have to collect molecules, existing molecules, which have shown toxic effects. Just like how we do in our drug discovery. You will have to feed in all the data. You have to collect about, for a particular toxic effect, collect about 100 structures or 200 structures, feed in the data there, expect the software to identify that structural error which is responsible for the undesirable activity. And then it will match with the kind of structure what you have fed as your degradant. And if it finds a similarity there, we call it as the similarity index, similarity, then it will disclose likely toxic effects for your drug. So it all depends on what is your input, the variety of molecules that you are feeding, which are reported to be toxic. You are feeding it, and then you are mapping it with regard to the structural entities present in your molecule. It's just in parallel line with what you do in drug discovery, more or less, if you have that knowledge with regard to chemistry, new drug
Yeah.
development. Yeah.
Yeah, so another question which is right now it is clicking my head is like when we do the force degradation studies, we
Yeah.
are deliberately increasing the acidity, the oxidative
Yes.
agent and
Yes.
all. So is it feasible or is it advisable to use those molecules for testing the toxicity? Because
Yes. Yes.
though it is
Yes.
as per ICH guidelines,
Yes. Yes. See, you are going for extreme conditions.
yeah,
Extreme conditions. That means you are trying to rule out all possibilities. See, when you
possibilities.
apply the extreme conditions, there are no other conditions remaining which can happen to the molecule to break
Yeah,
up. Right? So you are, applying those conditions, whether it is possible in the drug or not is a different thing. But its potential to break up when extreme conditions are applied, that means you are giving an assurance that this degradant will definitely not form, or this degradant will definitely form in some conditions in which that drug may experience from the environment. It is like that.
yeah. Thank you so much, sir.
Yeah. You can always write to me if you have any queries. I can definitely uh, enjoy responding to it.
This is very important topic, and we must be very careful while doing this type of work, because nowadays it is very much needed, especially in each and every API. We need to go for the degradation products and other things, and impurities especially. So that is very emerging topic, and at the institutional level, at least, we can do this type of work somewhat easily, although it needs some specialized instrumentation, GCMS and other things. But they,
Hello.
Yeah.
yeah,
Sir, this is Sandesh Ludha from Maliba Pharmacy College, Gujarat.
Yeah.
Sir, I have done LCMS for one of my project,
Okay.
but that won't suffice for mm, uh, the identification of impurity.
Okay.
So I was suggested that can we opt for, because they are not too much isolable.
Okay.
I cannot, the amount may be so less that
Hmm.
they are not isolable at whatever facilities I have.
Okay.
Some suggestion was there to go for LCNMR,
Yeah.
but again it's a too costly approach
Yeah.
for LCNMR. Can you
Yeah.
suggest anything else which can be done for? I have LCMS which has identified the impurity.
I understand from your query that the amount of impurity which is formed is below the threshold. That is expected to be, if it goes below the threshold, then you may not be able to detect that impurity. And I think you have got your MBI value for that. impurity?
Sir, I have that value. I have done LCMS, so
Okay.
that is not there. But still, the facility what I have,
Yeah.
I am not able to isolate it and give it for completely going for separation and putting it for direct NMR.
No, no. The otherwise, what happens? You see, with LCMS alone, sometimes you may not be able to generate the data what you are looking for.
Yeah.
Many a times, as far as I have seen, they put this data to LCNMR for purpose of confirming the structures. Because
Structure,
unless you
yes.
have a confirmed structure, you will not be able to use that data in order to predict the toxic effects accurately.
Yeah.
Because unless you know what is the pathway of fragmentation, many a times the pathway of fragmentation you will not be able to write unless you have NMR data with you.
Yeah.
So
So
many a times, many of the papers, when you go for presentation, I have seen reviewers inquiring about NMR data of these degradants in order to confirm the formation of, or in order to confirm the structure of those degradants.
yeah.
So
But
in your case, what I feel is, since you have not been able to isolate the degradant, perhaps the LCNMR only is probably the solution for it.
yeah.
Because see, the problem associated here is there may be many reasons why you are not in a position to isolate that degradant. That degradant itself may not be stable. That is the
Yeah,
biggest issue.
that can be.
Yeah, then that being the limitation of that degradant, I'll put it that way. The only other way is the moment that degradant is formed, it has to be fed into the NMR.
Oh,
And
but
huh,
yes, sir.
yeah.
Can you suggest where I can go for this LCNMR facility available in India, or
LCNMR
I?
facility is available if you are in touch. Are you in touch with any professors in NIPER?
Yeah, sir. I had inquired there only. Naipur
Yeah.
Mohali has it,
Yes,
uh,
yes, yes.
but it was a costly affair. So I just wanted to know
Yeah,
whether
it
in
is. It is. It is.
when
It is little
where
expensive.
whether
Yeah.
there is any other resource available.
Let me.
Right.
Yeah, you please get in touch with me. Let me just
Yeah, sir.
uh, inquire, and then I'll be able to let you know. It is an expensive. One thing I have to tell you is it is an expensive technique. It's a new technique. It's an expensive
Yeah.
technique. Yeah,
Okay, sir. Please do let me know your email
yeah,
ID.
yeah, yeah, yeah. You can.
Thank you. Yeah.
You are writing down
Yes, sir.
P for Pune, N for Nagpur,
Yes.
SANJAY
PNSANJY Sanjay
PAI
PAI.
at gmail.com.
Okay. Thank you very much, sir. It was
Yes.
a very well. I am too small to comment on your presentation, but it was excellent.
No, I just wanted to provide
And
you a bird's eyesight in 1 hour. What
yeah,
we can do in 1 week, I had to do it in 1 hour. So I don't know. But I have touched the critical points so that you know why this kind of study is required to be done.
surely, sir. Thank you.
And today you can see some things which we could not see earlier. No, just like COVID, we can see today. This COVID, even if it was existing 50 years ago, it could have just gone as cold and fever. I think that's all.
Yeah.
Hello,
Hello, tell me.
sir. Good evening.
Yes, sir. Good evening.
Yeah, sir. I want to add one thing for Sunday, sir. That is
Yeah.
LCNMR is available in the Pune University, sir.
Okay. It's available for outsiders.
Yes, sir.
That
Yes,
that
there is
facility is
a center. Yes, central instrumentation laboratory has been developed by
okay.
DST,
Okay.
and that for the inside as well as the outsider researcher.
Okay.
So anybody can come there and then they can run their sample on the hiring basis, sir.
Okay.
And the detail procedure how to approach for and how
Hmm.
to send the sample
Hmm.
detail is given on the website of the Pune University. under the heading of the CIF, Central Instrumentation Facility column.
Yeah. Okay.
Yes, sir. Yes, yes. So
Hello.
Loda sir can
Thank you so
contact there and
much.
you'll get facility.
Thank you, Vittal sir.
Yes, sir. Yes, yes, sir. Welcome, sir.
I am an alumni of Modern College of Pharmacy.
Great,
Oh,
Nice
thank you, sir.
Nice
Sir.
great,
Nice to see you, sir.
great cross interaction along with the interaction with the presenter. Any other questions from the delegates? So I can just, read out some of the reactions for Dr. Sanjay Pai. Very informative session, sir. Really, it was informative and resourceful lecture. Very informative, sir. And so on further. People have really appreciated your contribution and the deep insights you have given in the topic of impurity profiling as at various stages of the drug development. And the impurity makes the formulation or breaks the formulation. So it depends on how much impurity profiling is done. My question to Pai sir is, sir, as the impurity profiling is done more and more and the amount of impurity is reduced, the cost of the formulation increases. So at what stage the balance should be sought? Because the cost of the products in USA and all other European countries is higher, maybe just because of the impurity profiling.
Very thought-provoking question, madam. I have to put it that way. See, in the product development stage, especially in the product development stage, if you can foresee these kind of situations, what a particular drug is likely to face, then at the product development stage itself, the product manufacturing steps can be so validated to ensure that the product remains stable. Of course, we are doing a lot of work when the product gets developed, but then some thoughts are missed out when the product development is going on, like what are the environmental conditions that a molecule is likely to experience. We do apply certain levels of humidity, but certain issues like how the drug is likely to get exposed to, or where all it is likely to get exposed to till it reaches the cell. I'll put it the cell. See, there is a issue here also. When drug is administered, it may remain stable. But by the time it reaches the cell where it is required to get absorbed, it can undergo degradation even there somewhere. And then that degradant may form, and unfortunately that degradant, even though it may be present in small concentrations, today we know drugs which can elicit action even if they are present in microgram levels. And if this degradant is present in microgram level and has the ability to elicit unneeded activity, non-beneficial activity or undesirable activity, it is risky. So according to me, the cost adds on. It's a one-time cost with which I think the industry will be happy to absorb when the product is getting developed. So they have to ensure all possible ways in order to predict the possible degradants which are likely to form before the launch, and perhaps once they validate the method appropriately, it should not cause much of issues, many of issues, at least predictable issues, so that the costs don't add up. I'll put it that way.
If I have to summarize what you say, I think you are propagating the concept of quality by design.
Yes, 100%.
So quality by design, if we take care of the quality at each stage and avoid entrance of the impurities at various stages or decomposition, then we can reach a better product.
Yes.
Thank you so much, Dr. Sanjay Pai, for your valuable contribution and the deep insights which you have given to the people. Like today's day, we started with, drug development, and then drug development comes to formulation and then to the analysis. So you have,
Yes.
towards the end of the day, it is rightly gone, starting with the, design of the drug to the analysis of the drug and impurity profiling of the drug. Thank you so much.
Okay.
So I request Dr. Bakode to present a bouquet of flower to Dr. Sanjay Pai. Sir, kindly accept the virtual bouquet, sir.
Thank you, madam. Thank you so much. Yeah.
And, I have to present the certificate as well.
Yeah.
Again, virtually we have to present you, but this certificate would be sent to you by mail.
Okay.
So kindly accept the certificate as well.
Yeah. Thank you so much. Yeah.
Thank you for sparing your valuable time and giving the knowledge to our delegates. Thank you so much, sir.
Thank you, madam. It was pleasure, being here online and share my thoughts with the delegates of your program. I think I wish all the very best to all the participants, and I think they will have many more things to take away, to carry away at the end of the program. I think which goes right up to Saturday. Best wishes for everybody, and I think.
Queries and everything is remaining. They need to come to the Goa to meet and consult you.
Yeah, welcome. You are always welcome.
They would be too happy to go to Goa. In addition to meeting the sir, there are so many more attractions at Goa.
Yes. We only are waiting for this pandemic to subside.
Yes, sir.
As early as possible. Yeah.
Right.
Thank you very much.
Thank you, sir.
Thank you, madam. Yeah, thank you so much.
Our next speaker, I have to just check whether he is on board.
Take 5 minutes he'll taking.
Achha, he'll take 5 minutes to join. Okay, fine. No problem. We have to get introduced to our delegates. Yesterday we had introduction of few of the delegates, but we could not complete it. So now I request few more delegates to introduce themselves. Can Babita Agarwal introduce herself?
Yes, ma'am, my pleasure. I am Dr. Babita Agarwal. I am an associate professor at Marathwada Mitra Mandal's College of Pharmacy, Thergaon, Pune. And I've been working in this college for the past 12 years. I have my specialization in pharmaceutical chemistry, and my basic interest, like it is more towards the analytical section of multi-component analysis and these sort of things. The very important thing I would like to tell you, ma'am, that I thank this COVID situation. Otherwise, I would not be able to attend a QIP program of DIPSAR. Due to this COVID situation, at least I am able to, we all are actually able to gather so much of knowledge otherwise, which we would be deprived of. Thank you so much, ma'am, and all the best for all your endeavors and your college's endeavors, university's endeavors.
Thank you, Babita.
Thank you, ma'am.
Great introduction. All the best to you. Can
Thank
I
you,
request Tejasri Sande?
Hello.
Tejasri.
Hello.
You are audible.
Am I audible?
Yes, you are audible. You are visible.
Okay, ma'am. So I'm Tejasri Sande, and I am working as an assistant professor in Rasiklal Dhariwal Institute of Pharmaceutical Education and Research. I'm here for 6.5 years now.
Which place it
really
is?
6.5 years experience in teaching.
Which place your college is at?
Pune.
Okay.
Pune. Okay. And I'm also thankful to this COVID situation because this is my first QIP program. And as this is online, I am really able to join this program, and this is really informative session. And thank you for all you faculty, and you are really conducting very good. So thank you so much for the all you DIPSAR.
Thank you, dear. Can I request Adil Ahmed? Is Adil Ahmed there? Can I request Ajay Sharma? Is Ajay Sharma there? Atul Khadse?
Ma'am, excuse me.
Yes.
Ma'am, I would like to tell one small thing. Dr. Ramesh Goyal, when I was studying in D.Y. Patil College, we had a guest lecture of Sir. And I was so impressed by the way he was. I don't exactly remember the topic, but जो बोलते ना उनके तेज जो था ना उनके चेहरे पे, it was like, oh my god, even we can do so much things, so many things in pharmacology, but somehow I was scared of animals, so I couldn't go in pharmacology. And whenever you meet Sir, please tell him that he has influenced many people's life by giving his guest lectures in our college.
We will surely tell him, there is no doubt, Babita. In my mind, he is no less than an excited electron, you know. I always take him as an excited electron, always ready to go in the higher orbital. So his energy is so much so that we get impressed how he can manage at this age so much work. Ask him anything, anytime. Like, for example, yesterday afternoon, you all must have noted that there was no speaker that moment. At last moment, the speaker denied. And casually it was asked and he started. So it is a motivation to all of us. Frankly speaking, because you have taken out the topic, my feeling, I'll tell you, during my growth years, I always feel if I would have had a boss like him, I would have really grown much, much more than what I am today.
Yeah.
Because he sets the example and always motivates.
Yeah.
Great, great. We will surely convey. Can I request Subhash Deshmane, sir?
Yes, good afternoon and namaste to all of you. Good afternoon, Parle Madam, especially Wakode sir. Myself, Dr. Subhash Vishwanath Deshmane. I am working as an associate professor in Rajashree Shahu College of Pharmacy, Buldana. My specialization is in pharmaceutics, and still I am interested to join this QIP because already 15th QIP I have joined at DIPSAR in 2011. Wow. Yeah. Thank you so much, and thanks for nice conducting the program. Even this is an online or virtual program, but still conducting in good manner. And we all are very convenient to this program.
Okay, tell me one thing, because you have attended some 15 programs at DIPSAR. Do you feel physical mode of the program better or online mode?
Okay. No, definitely physical mode of the program is definitely better than it's online. Because real interaction with all these speakers.
All good. Okay. Bye.
At the DIPSAR or even in physical interaction as compared to the online interaction.
Okay. Can I request Ritesh Popat to say a few words?
Amrita Madam, excuse me. Amrita Madam.
Yes.
I would like to few sentences in addition of the Babita Madam. She told about the Goyal sir, but I would like to add.
Please introduce yourself. I am unable to understand who is speaking.
Madam, I am Vitthal Chopde. I am Dr. Vitthal Chopde from Modern College of Pharmacy.
Okay, can you put on your video, please?
Yes, madam, my video is on.
Okay.
Audible?
Yeah, you are visible.
Yeah. Madam, I would like to thank to you also, especially as you and Pakode sir are our senior teachers, still your enthusiasm and your energy is very high than us. So you are organizing very nicely. I'm watching to you from morning to evening, but you never see as you are tired or you are taking the shortcut way or to wind up the close. I never seen that kind of things. I really thank you, Amrita madam, to you and Wakote sir. You are really very enthusiastic. We are proud to say that you are our teachers during our college. Madam.
Thank you. Thank you so much for your kind words. And
Yes,
Dr.
madam, yes.
Wakode also would agree that you are giving more credit than what we deserve. Thank you so much.
Yes, madam. Yes, yes.
Our next resource person is ready now.
Ready?
Yeah.
Right. We will continue with the introduction. In between, as and when we get the time, let us go to our next talk of the day. It is the fourth talk, and we have the speaker from United States of America. He is a global head. He is a global head, chemical and pharmaceutical profiling at Novartis, USA. He is going to speak on drug discovery and development in pharmaceutical industry. Dr. Sudhakar Garad has received his PhD from Bombay College of Pharmacy, University of Mumbai in 1998. Sir, Garad sir, we are alumni of the same organization. So I really feel nice to read it. He has worked under the guidance of Dr. Nagar Senkar. Soon after completion of his PhD, he worked for a small stint with Pfizer in clinical research. And because of its, for his extraordinary credentials during his PhD, he was selected as a PDF postdoctoral fellow at University of Connecticut. He has worked on microenvironmental properties about dissolving surface of the polymers under the guidance of Dr. Robin Bogner. After his PDF, he has worked with Vertex Pharmaceutical for 3 years as a senior investigator in formulation development group. After that, he joined Novartis as a group head, and he worked with Novartis for a long period of 9 years. He joined as a director of formulation development at Cubist Pharmaceuticals, or it is known as Merck, for 2.5 years, and currently he is working as a global head of chemical and pharmaceutical profiling, discovery of pharmaceuticals and novel drug delivery systems. He is also disease area head at, especially for the new indications, at Novartis. His primary role is to collaborate with his research colleagues for building the right pharmaceutical properties into new chemical entities. He does that via solubility or dissolution enhancement technologies to expedite the molecules into toxicological and clinical studies. He is responsible for enabling research, clinical and commercial molecules via novel drug delivery technologies, especially brain targeting, permeability enhancement, TI improvement. I do not know what TI means. Dr. Garad himself will tell what TI is. He has many publications, book chapters, and patents. In last 20 years of his career, he has taken, this is very surprising. In last 20 years of his career, he has taken more than 200 chemical entities in clinical studies across a dozen disease areas. Just imagine working on 200 chemical entities for more than 10 disease areas. That is something very creditable, Dr. Sudhakar. It's a pleasure to have you in DIPSAR in the 32nd Quality Improvement Program sponsored by All India Council of Technical Education. Welcome, sir, and the desk is to you.
Thank you so much for a kind introduction, Farley Madam. It's an honor to be here and be with you early in the morning. And sorry, I missed a couple of early lectures, but now it's 6:30 in US. A very kind introduction. Appreciate that, and like to thank you, Sharad, also. Sharad, Professor Sharad Vakude, you might know or may not know. He was my roommate, we were classmate back home, 1989 to 93. It's like a brother to me.
Yeah.
And when he asked me, Sudhakar, is it possible for you to give a lecture? And I said, it would be my honor to, share my knowledge, with professors, because you are the pillar of, of course, academic, and I'm not able to, I won't be able to teach, but share my knowledge with you. So with the, with you. So I'm trying now. I'm trying to share my screen and having slightly difficulty. I don't see the option to share the screen somehow. So just clicking. I see.
Are you able to do it, Sudhakar?
Somehow I'm not able to see the screen. Let me quickly log off and log in again, if you do not mind, just for like a second, okay?
No problem, but I think a screen share option is in between, and we have allowed it. Just check it once.
Yeah, I'm just trying to see, because if there are I can still hear you, Sharad. Although it looks like I logged off, but I can still hear you. Can you hear me?
Yeah, we are able to hear you.
Yeah, let me exit, if you do not mind.
You can see here Dr. Rajesh Oswal ji. Can you introduce yourself? We are seeing here regularly, punctually attending all lectures. Dr. साहब, आप just introduce कर सकते हैं? Just unmute yourself. Sir, unmute कीजिए.
Hello.
Oswal साहब, we are unable to listen to you.
Sudha. Acha, एक minute. Dr. Sudhakar, can you hear me? Dr. Sudhakar, can you hear me?
Yes, I can hear you. I can hear you now.
Sir, what is your name in the Zoom?
GARADSU.
GARADSU.
Hello.
Hi there.
Sir, now you are co-host and you can share the screen. Just try, sir.
Yes, I can see perfect now. Yes, thank you.
Okay, thank you.
Now I have access. Thank you so much.
You have not started your video?
Yeah.
Yes, we are able to see the things.
Can you see my screen now?
Yes, sir. We can.
Very nice. Very nice. Apologies for technical difficulties.
Right.
Somehow I was not able to get that option, but now I do.
So we welcome you.
Thank you. A very nice introduction, Parle Madam. And without further delay, I will keep going. Thank you, Professor Wakode. Thank you for giving the opportunity and a very nice introduction, Parliament.
Welcome here.
With that, today what I thought was, why not give an overview on drug discovery and development in pharmaceutical industry. Especially back home in India, there are, I think, half a dozen industries are getting into basic research. I saw Dr. Reddy's, Glenmark, Lupin, I think Cadila. There are very few of them are working on certain targets, especially in cardiovascular diabetes and inflammation in those areas. But majority of industry is generic, right? Most of the industry over there, generic company, where they really do not have to do basic discovery. And and the whatever products we have, as you know how generic operates, I won't go into detail. So that's the major focus back home in India. However, in Europe and US, majority's focus is on drug discovery to invent a small molecule or large molecule. My experience basically is in a small molecule, new chemical entities. All my career went in small molecule, or new chemical entities. Biologics, we do have biologics, but my expertise are not in biologics, only small molecule. And as you know, biologics are very expensive. Biologics such as protein, peptides, antibody, those are biologics. And usually they are very expensive. And there are not many products in the market because they are, not because they're expensive, but one of the reasons they are expensive, it takes longer time. And the targets in the human body are usually very small. So if you have to get the efficacy, then usually majority of the time you get it with the small molecule than large molecule. But many companies are working on large molecule, just to get an idea where this is coming from. Okay, so with that, I'm trying to see a screen. Okay, so I'm clicking on screen. Someone wants to join. Okay. Okay. So you see my screen now?
Yes.
Okay, good. So
able to see nicely, sir. No problem.
Okay, good. So sometime I'm not able to, see screen going up and down, but that's okay. Now I can see I think it's a second slide. So this slide suggests that, as you can see, how the drug development process, happens. It's kind of traditional approach where you see my cursor as well.
Yes, sir.
Okay, good.
Yes,
thank you. That's important, so that I
see the cursor as well.
Thank you, ma'am. So you can see from overall from selecting candidate or identifying candidate to all the way up to commercialization, from FDA review, large scale to commercialization. You could see it takes from 9 to 15 years. There are sometimes situations where within 5 to 7 years also you could launch product, but usually between 9 to 15 years. And why it takes that long, and we can go over very briefly. You can see in drug discovery, when you call drug discovery, you can see there are a number of steps, and there are on other slides I have went into detail, which will go there. But here you could at high level you could see there are 5 to 10,000 compounds are being screened over here, from molecular modeling, to synthesis, to doing in vitro testing, and then from there you can imagine from there around 250 molecules further, because it's kind of funnel. It filters the molecule depending on number of criteria, and then in preclinical we would see around 250. Now what happens in preclinical? That also we will go into more detail, but majority of the time, of course, pharmacokinetics, pharmacodynamics, pharmacology, mini toxicities. These are the major things, three major things: PK study of a compound, pharmacology of a compound, and safety of the compound. Those are the three things happens in preclinical. And when that happens, when the molecules passes those hurdles, we have excellent PK, we have good pharmacology, and molecule is safe. I mean, safe in the sense it has a margin. So the madam was asking what is a TI? TI is a therapeutic index. So so if you have that's the difference between pharmacology dose and toxic dose. For example, your tox dose is 100 milligram, and your efficacious dose is 10 milligram. So you divide 100 divided by 10, then it comes 10. So 10 is your TI, therapeutic index, which is very narrow actually, because if somebody takes 2 or 3 pills, then it's little, just by mistake or, or forgot to take. So usually when just about the therapeutic index, as you know, pharmacology teacher, of course, they know TI, I'm sure. But as you know, the TI has to be really, really high means 100, 500, 1000 therapeutic index, because that gives a broad safety margin, in nutshell. Molecules such as molecule for blood pressure, molecule for diabetes, or in nutshell, molecules which we have to take throughout your life, or our life. There are certain diseases, you have to take medication rest of your life. For example, blood pressure is being detected, diabetes is being detected, if it happens. So for such medication, for such indication, you have to take medicine rest of your life. And there you need to have margin of safety, which is TI, very high, 500,000. That's the significance of TI. So over here, when those molecule, like in oncology, for example, little bit about the therapeutic index. In oncology, as you know, it's very difficult to discover a very safe molecule, because the way oncology molecule or cancer drug, the way they work to cure the disease, or the tumor. And those who understand the pathophysiology of cancer, so normal cell also divide and multiply, and cancer cell also divide and multiply. Now cancer cell, they divide and multiply at very high rate compared to normal cell, and that's why there is a tumor growth depending on the kind of cancer. Now it's very difficult for scientists to target specifically a normal cancer cell, right? I mean, both are the cell, right? And how do specifically target the cancer? So that's a challenge, and it's still a challenge. Actually, there is nobody who's, of course, because of the mechanism. So it's when you try to have affinity of our molecule, of our drug, new chemical entity towards cancerous cell, they do impact a little bit normal cell also. So in nutshell, for oncology, we are talking about the TI, the therapeutic window is really narrow. They will be very happy if the efficacious dose and toxic dose is more or less similar. You can imagine your tox dose is maybe 10 milligram, your efficacious dose maybe more or less 10 milligram. You can imagine that is the situation. And when somebody goes through cancer treatment, you see the situation, loss of hair, chemotherapy, weakness, other side effect, until the treatment is going on. And as soon as you stop it, then you recover back. So, okay, so in on this particular slide on preclinical, when you clear that hurdle, then you enter into phase 1, phase 2, phase 3. And as you know, majority of the time, you cannot skip all these phases. You have to go through a number of phases, and you can imagine the number of volunteers. So in phase 1, usually they are healthy volunteers, except oncology. Because cancer drug, as I just said, I just told you that it has a narrow therapeutic window. So you really cannot test on the healthy volunteers because they're oncology drug, right? So they go directly into patient. Those who do not know, oncology drug have to test directly into the patient, cancer patient, not in. But all other indication, I would say, majority of the indication, other disease area, you have to do phase 1, single ascending dose, multiple ascending dose in healthy volunteers, and then move on to phase 2. Right. So once you finish phase 1, phase 2, and again, number of volunteers, that depending on the indication, that depending on the target. If there are only 1,000 patients in the world, you don't need those volunteers, right? But cardiovascular diabetes, the number of volunteers are really, really high. To just get an idea. Now, once you get all this data, right, then you file NDA. You submit NDA to FDA. And of course, they usually take 0.6 means half a year to 0.9, less than a year to review. And sometimes if documentation data is not satisfactory, what they are looking into, then they have to ask, come back. So it might take close to 2 years also, but usually 0.6 to 0.9 years. Okay, and once they approve and it gets into the market. Right, so that's just I'm talking about how traditionally drug development, oh, so you are not able to, I'm sorry, I thought you may be able to see my video also or me also, but I just realized that you were not able to see me while I was talking. Apologies. But now perhaps you can see me and you can, okay, good. So on the next slide, I see a lot of colleagues are joining in between, so I'm admitting them. So
We are doing it.
Okay, good. So we'll talk about the cost, right? We talk about the time, how long it takes. We said around 9 to 15 years, and now we talk about the cost. Cost varies actually, and then you see in summary, I said from 1.5 to 5.5 billion. So depending on the small molecule or large molecule, depending on the patient population. Usually, as you know, in majority of the country, if your new chemical entity has to launch, we need to do clinical trial. For example, if we would like to launch product in Japan, so we cannot conduct all the clinical studies in United States or in Europe or anywhere in the world and just launch the product in Japan. We can't do that. That's usually against their country's policy. Japan is particularly very strict. They even preclinical study, if you are conducting phase 1 studies there, phase 1 studies, some people are waiting in the room. I'm just trying to allow them.
We'll do it as we have given you host one. That is why it is you are able to see it. But we are admitting.
But you could do that. Okay, I don't need to worry about it, right? Yeah. Okay, all right. Thank you. That makes my life a little easier. Thank you so much, sir. So there we have to conduct the clinical studies. So again, depending on, as I said, patient population, it end up in costing from 1.5 to 5.5 billion US dollars. So you can imagine how much cost we spend launching a new product, right? Or one product. And that's why the patent plays critical role. As you know, you might hear like big pharma invent molecule, but nobody can launch other than them because they are first inventor and they invest a lot of money, so they own that. Now we'll talk about slightly little bit about patent. Those who are not aware, although I haven't highlighted here, once you discover a molecule and go into preclinical studies, it is usually safe to file patent before your phase 1 result comes out. So that way it is very easy. Easy means nobody will have access what exactly you are doing. Until then, it's better not to file, because the minute you file a patent, it starts actually timing from that day. So time start clicking. Suppose you discover a molecule today, 2020, September, and if you file patent and it gets granted within 2-3 months, and you have 17 years, 17 years on that particular molecule. And I was just saying that it takes from 9 to 15 years to launch the product. So if you file patent now, and if you take 15 years, you have only 5 years to cash, or to milk the cow, kind of, It's just only, you have only 5 years. So if you put 5 billion and you are just going to get benefit for 5 years, it's for patient actually. Usually in pharma industry, most of the pharma, their mission is to how can I discover, develop a molecule, to save and extend patient life. That's their mission. That's everybody's mission. And that's what motivates everyone, including myself, because you're working hard to save patients' life indirectly. However, when you work hard, we need to have, returns. So my point is, if you file right away, then you have only a few years to, access or have access to that patent or that molecule. Therefore, many pharma industry, what they do, as I was just saying, that at the end of phase 1, it's official, right? The data they may not be able to see, but you file on FDA site, you publish. So many competitors come to know that, oh, you have this target and you are working on this and you have phase 1 results. Then everybody starts working. So then in order to save your asset or that particular pattern, you should file by end of phase 1. Or as soon as you file the NDA, you should start filing pattern. And then starts clicking the time. And that's why many pharma, and you will see on the next slide, what I'm trying to see, because it's because of the, you have only 17 years, okay? So you got an idea, the cost and how much time do you have. So now at a very high level, it's very important to understand, that we usually do a lot. As you saw the discovery, like 5,000, 10,000 molecule early on, as you saw in the previous slide. But then what is the success rate? That is another factor. In industry, scientists work really, really hard, but success rate is not that great. You can see over here, only 56% molecules are successful out of 100. Just an example. Then you go in phase 2, only 20%. Then only 12%. And at the end, if you have 100 molecule, only 11 molecule we were able to launch. That's really high actually. So in nutshell, it's only 10% success rate, right? I mean, imagine if you put billions of dollars in inventing molecule and your success rate is only 10%. Will you do that business? Especially back home in India or any other country? Well, say, कुछ फायदा ही नहीं है यार, इतना पैसा डाल रहे, कुछ भी नहीं मिल रहा है, चलो हटा दो. Or you have to wait for 10-15 years. I'm just bringing a little humor. And this is not how, big pharma or corporate pharmaceutical, they think. We all know the success rate is less, but if we don't do, who will do it? All this new chemical entity, as you know, who will discover? So we have to keep in mind why it is expensive, why medicines are, why it takes too much time as well as cost. And why the medicine, when the big pharma launch, you must be hearing, oh, this molecule being approved, treatment per year is $35,000 or per month is $25,000. The reason is, and most of the time insurance do cover those costs, but just to get an idea, these are the reasons behind why they are expensive and why it takes cost. Okay, these numbers are a little bit older, but I think you can imagine these numbers are in millions. But again, this is a rough, very rough estimated cost. All right. So now we learn about the time, we learn about the cost, and now as you learn about the patent situation, and that's why most of the management colleagues they usually say, hey guys, well, the results are out now. Can you shorten the timeline and reduce the cost? This is usually project management. Project management play a key role to reduce the timelines. And that's why I wanted to highlight it. Along with the science, the main point of this is along with the science, as you see, or you have seen, the project management play a key role. Those who are from industry or have joined industry and came to academia, or your friends are in industry, you might know there are project managers, in industry, and those play a really critical role in reducing time and cutting the cost. Okay, that's the only essence of this slide. Now, you see on the another slide, what are the different project phases, in discovery. And this gives really detailed explanation. I do have a copy, PDF version given to Sharad sir, and he will be happy to share with you, or you might have it. So I'm not going to go into more detail because you can see there is a lot of description over here. But I will give you an very high level at each bullet, what's happening. In exploratory, early on, if somebody says I want to find a target, so it's usually you can here you have to explore the biology of the target, its pathway, what is its relevance to human dose, and validate that target on computer or with the preclinical. That's how exploratory, just identifying target, and it takes 6 months to year to 2 years. It's not that easy. Identifying the target, purifying the protein, doing a single crystal, identifying that particular protein structure, and then do molecular modeling. Those who are in a CAD, or you must have heard the talk, or you will hear. It's not that easy, it's not that easy means, let me correct. If you are, and we might have another slide, and we'll go into more detail. But if you are first in a class, you are the inventor. Then it's not easy. If somebody has already identified and you want to best in the class, then you can take everything from them and keep doing. So for the inventor, it's not easy. Okay. Then in drug discovery, you can say, here it's important to do number of assays and compound. I won't read it, but you get an essence. You can see into more detail on the slide. Now, during optimization, this is very important. Actually, this is the phase where my team involves. You can see that what Sudhakar does in Novartis. This is where my team involves, where optimization. Now, you see here a small development candidate. Now here we are able to select the development candidate out of 10, out of 100 for clinical studies, and where my team is heavily involved in selecting those candidates. Now, one thing I could proudly say that why so early? Here, what we do is early on, we provide formulations for pharmacokinetic, pharmacology, and tox studies. That's the one, because we are a formulator, mainly drug delivery formulation group. We have 300 targets. We have 5,000 research colleagues. And for them, my team, close to 100 colleagues globally, between US, Switzerland, and China, we support those preclinical formulation studies. Now, most importantly, along with supporting formulation, we are a kind of a value-added team. Now, what does that mean? And I'll just give a brief example. For example, you have a molecule coming from research has 10 microgram per milliliter solubility, just 10 microgram. That is not going to help for preclinical formulation or clinical. So what do you do then if 10 micrograms? Sometimes you have to develop. We have technologies to develop to overcome those solubility. But then we need to understand the kinetic solubility. So if kinetic solubility is 100 microgram, then we can use different technologies to make that successful. Now you must be saying where these 10 and 100 micrograms numbers are coming. So after like 15-20 years of time we put into 100 microgram per ml at physiological condition, its solubility is good for preclinical as well as clinical studies, which gives us if it is permeable, molecule has to be permeable, gives more than 90% fraction absorb, then you have almost 90% to 100% bioavailability. If it doesn't metabolize in liver, that is very important. So we kind of give feedback to have 100 microgram solubility in their molecule. Now, one is giving feedback and having guidance, or tell them to have this one. Secondly, when our colleague sat in research team, or when we are with them, we try to say that how about adding some functional group, ionizable group, solubilizing group, where we could make easily a salt or co-crystals, so that we can improve the kinetic solubility dissolution, and which we can maintain by certain polymer or surfactant. So this is little basic science. What I'm trying to say is my team involves here, and we give intellectual input during research to have a right biopharmaceutical properties in the molecule, so that during development the solubility dissolution are no more challenges, because many molecule fails because of poor exposure or poor dissolution. That's my team does. That's my one. And then we enter into the preclinical. Okay, preclinical stage. Then from preclinical on next slide to highlight. Sharad, I don't know how much time, but I will try to speed up.
15 minutes. You can.
Is time is a constraint, Sharad, or we are good?
10-15 minutes.
Okay, good. Thanks, sir. So then from preclinical, as you saw, we enter into the clinical, which I gave a really good description on the first slide. What are different phases and what not. So I won't go into detail, because in phase 1, in phase 2A. But one take-home message is that whether your molecule is working or not in a human, that is studied between between phase 1 and phase 2A. That's called proof of concept. There we really understand molecule is working, and once it works, then in phase 2B we have 1 or 2 doses, mostly 1 dose, but depending on the indication and the target population, maybe 2 doses in phase 2B, but usually 1 dose, because phase 2 studies are the studies where you will enter into phase 3, and phase 3 studies, clinical phase 3 studies, are more or less commercial, commercial dose. So depending on whether you could have 3 doses or whether you could have 1 dose. So my point is, usually 1 or 2 doses are being studied. In phase 1, there are multiple doses you have to study. There's no doubt. But in phase 2B, only 1 or 2, and then those doses are continued in phase 3, just to take home message here. And number of patients are high also. And then, of course, as I said, in outcome of phase 3, you file registration, and then it go for in post-launch also we do a lot of studies, but that's then understanding what is the impact of food, what is the impact of other medication, and then you come up with, like, do not give with this, do not during this phase, from phase 1 to phase 3, we do understand a whole lot the drug-drug interaction, but most of the drug-drug interaction studies we do post-launch to make sure that these drugs are being given or not given. But very commonly used drug, usually we have to study during phase 3 only, because we have to understand the impact on absorption, on the pharmacokinetics and whatnot, just to get an idea. All right. So now moving on to next slide. Now, how do you select the project, right? Just to get an idea, we heard about how long it takes, how much it costs, what are the different stages are involved in drug development, in clinical trial. And the basic parameter is, if some professor would like to, especially medicinal chemist professor, if they say, okay, maybe I might have a better scaffold, better molecules for certain indication, and I would like to form a company or partnership with someone. For those, it could be very helpful, this one. So overall mission, it's very important to understand what is our mission. Do you want to be in 10 disease area, or you want to be just focus on certain disease area? And in Boston, there are 600 biotech companies. They will not work on 10 disease area. They will work on one indication, one target, or maybe two target, and that's it, and try to come up with the molecule. And we need to have right expertise. That's very important. Somebody has worked in cardiovascular all his life, inventing medicine. Suddenly you cannot hire that medicinal chemist for oncology. Chemistry background remains the same, but biology part is missing, and you cannot invent medicine without knowing the biology. So the hiring person cannot say, "Arey, kya ho gaya yaar? Tune to cardiovascular mein kiya, chal aaja oncology mein." You cannot do that. You could do that if he's your friend, but you will not be successful. So you have to really understand. So that's why I think right expertise at the right time is the key. No more favor, no more okay. Oh, mera dost hai, oh, it doesn't work that way here. If you have to really bring medication to save patients, you need to have the expertise right area. Someone might say he might have good managerial expertise, and as a manager you are okay, because what you are doing is you are giving complete freedom, access, empowering, the flexibility. That environment you are creating, it's perfectly fine. But if somebody has to work hands-on, you need right expertise at the right time. Third point is first in class or best in class. This is I didn't want to explain on previous slide. This is very important. Do you really want to be the inventor, which nobody has worked on this target? Then you are first in class. You are the one. Best in class is somebody has already invented. They are in clinic or in the market. Now you want to be better than them. Then you are best in class. At Novartis, where I work, we are 90% first in class. We always have molecules or targets where first in class, so nobody can touch it. Of course, in preclinical there might be some other players, that's a different story, but we always want to be first in class. So which means time, speed, your dedication, passion are the key role, the people who are working. That is very important, and that's why I'm saying it's validated target versus novel target. So validated target for best in class, novel target for first in class. All right. And now whether you want really a small population, $100 million product, or you want blockbuster, $1 billion to $10 billion. That is one of the core mission. Some people, they are caring about patient. They do not care about whether it's 100 million or 10 billion. I want to recover. I want to cure a patient. That is the key, right? And they go after this mission of bringing a new molecule, new medicine into industry, not worry about but some companies say, what, our goal is to bring blockbuster. We don't want to touch 50, 100 million because we are big engine and we can't feed our all associate with 100 or $200 million commercial market. We want blockbusters. So that's one of that's as a management you need to decide. Develop and commercialize by yourself, or you want to out license. So small company, I was just giving example. If some professor wants to have a pharma, a biotech company, and what they could do is they could invent molecule, they could bring it to talks or up to phase 1, and then they could out license to big pharma. All the big pharma, or maybe I can give one example. You may or may not know a company called Celgene. Celgene was bought by BMS for $100 billion, and 75 or 100, I don't remember exact number, but nonetheless 75 to $100 billion. Now Celgene was an oncology company. Purely they operated by buying asset from small pharma. What they did was they had a small invention, but majority of the molecules were coming from buying or in licensing. So they would in license, and then most importantly availability of the funds. Right, fund has to be there to make this happen. All right. So first in class, very briefly, I can tell you that first in class, you have 100% market, no brainer, right? Because you are the one, nobody's there, so you will capture 100%. But you are best in class, somebody is already in the market and you are a copycat, your market share is going to be not more than 40%, unless there are a lot of disadvantage in the front runner and your molecule is overcoming those, then you may be able to go more than 40%. Usually doctor loves inventor and they don't prescribe the copycat. They do, but not really. That's why that's why majority of the big pharma, they go out for first in class. Okay. And then see the first slide when I said traditional drug development process. Now what I'm highlighting here is the new trend is now. Remember this? You see these boxes, lead optimization, this a number of different phases. We can't eliminate those phases due to Corona. Now FDA is also thinking that if there is a possibility to jump from phase 2, phase 1 to phase 3 with the sufficient data, you could do it, but no shortcut. But depending on the target and indication, you could do it. That's why the Corona vaccine vaccines usually won't take that long. Usually 4 to 5 years for vaccines, proper vaccines. But then because of this situation, in less than 1.5 years, I would say by early January or April, we should have there are 5 players now in phase 3. The outcome should be available end of October, November, and then it's just the manufacturing. But in 6 months, they have to monitor the side effect and the antibody response. FDA will not approve right away. But if this is the emergency, they might take a little risk, but 6 months is always follow up to see the safety as well as the antibody immune response. So they might come faster. They will instead of might, and they must, because we are all suffering very badly. But usually now what they call is exploratory phase. You are exploring because you are exploring multiple molecules with the proof of concept. And once you explore and identify, you confirm in broader population. So exploratory and confirmatory phase. So with this, we could have a shorter time period. Okay, just to get going. At very high level on this slide, what I said was, big pharma, you can imagine how many indications they can have, and then the percentage of, so, but the small biotech, they can pick up only one disease area and they can work on that. This is just to give you an idea how this happens in big 10 multinational pharma. If you take, this is the usually model. They work on more than 300, 400 targets in 8-9 major disease area. You can imagine 8-9 disease area, 300, 400 targets, and you spend $10 billion every year, and you might get 2 proof of concept, and then you move on to the next study. So you can imagine the cost involved and how much, So this is just to get an idea in project selection process. There are a number of pathways. In oncology, there are 400 pathways. I'm just highlighting the number to get an idea. Now, which pathway you have to pick up, which target you have to go after, that's you and your team should decide. Where is the likelihood of success? So drug discovery is not an easy. My message here is it's not really easy. If you have a 400 pathway in oncology and you go for particular indication and then which one to pick up and where the safety is important and the likelihood of success to treat or inhibit that disease is key. So this is very high level to understand how many pathways and what disease area. Now when I was talking about the expertise, which was very important, right? I was saying that you need right expertise as right people. So you can imagine how many different functions are involved in research science, from translational medicine to preclinical safety, in proteomic chemistry. And when I was saying proteomic chemistry, a little bit like identifying the structure of that particular protein or target and see here how your molecule fits in. Do you get that affinity, selectivity? Is it hitting other targets and whatnot? This is, so you can imagine, and then of course, DMPK, ADME, analytical sciences, chemistry plays a key role because they are the one who will discover the molecule, right? They are the one who will identify. And these are all other, more or less, you can say supporting function, but instead of supporting, they also give a lot of intellectual, as I gave example of my team also, how we give intellectual input during discovery, because once molecule is selected, there is not a whole lot you can do. Prior to the selection, you could do as much as you could. Spending 3 months extra on having right ionizable group, having right solubilizing group without impacting its efficacy and pharmacokinetic, if you could, then better to select that molecule rather than later on you playing around. You know? So, you know. Now, after knowing that when you have different line functions, what I'm trying to highlight on this slide is how the management, of a project by a project manager. You know, so project manager could be within a line function, between the line function. He or she is a leader. You know, from each line function there is a representation, right? And then of course what they do is they follow regularly. There are meetings. You might hear like, oh. People are always in the meeting because they have to understand what other line functions are doing and what they need and what your line function can do. And that's how the teams are being formed and projects are being involved. This is just to highlight that in exploratory phase, what different activities are done. This is a repetition of slide. It's just how project management. This slide is kind of separating the project management. You have project management prior to the proof of concept dedicated, and then you have project management, after the proof of concept. And this slide gives you a very high level idea how different line function from PK science, development, their short form, technical R&D, if you see my cursor, preclinical science, translational medicine, drug regulatory affairs, different line functions have their timing. So everybody is on the hook, right? Everybody has to deliver. So every line function is on the hook. Everybody has to deliver. They are connected to each other. And this is how the drug development path process happens. Just wanted to give you flavor on the timeline, how this complex process the pharma industry must be managed. This usually taken care by project management. He or she is responsible for timing and for the duration and the activities. Line function will do the activities, but what activities? For example, my line function, solubility, stability, physicochemical tox formulation. This is my line function, which is TRD, technical research and development. And a lot of people depend on my activity because we are the one who gives the formulation. You can see formulations for DRF. So they are all interconnected. Okay? Just to give you a flavor. And now it's a development. So now when a proof of concept is happen, then project manager changes because their expertise up to research, right? And now development is a different phase. So there we require different project managers or different expertise to the commercialization. Okay. And in development science, as you saw in research, how many colleagues are involved from different line function. Now in development science, you can see translational medicine, preclinical safety, marketing, technical R&D. We play critical role because you are drug substance, drug product, quality assurance, quality control. You just name it, drug supply, drug substance manager, drug project management, whatnot. Clinical team is one of the, every line function is the key. Without drug substance, drug product and clinical team, nothing can go. But then regulatory colleagues keep an eye. You just can't put anything in the clinic. They are the one who, maintains along with the quality. So in summary, you can imagine different line functions are involved in a drug development. And that's why in big pharma, you will see so many associates. And that's why so many associates are required to make drug development process. All right. Moving on to the next slide. Just to highlight that during development, same slide previous to, within the line function, between the line function, and there are different action item to follow. So very briefly, wanted to give you a phase 2, phase 3 example. You can imagine over here, if you say I want to go ahead and in phase 3 right here in Q1 2021. And those who are involved in the drug, they will say, okay, you really cannot do, what you have to do is this FMI means market image formulation. Market image formulation and lab phase, we need to do those studies and we have to have a pilot phase. Once we confirm all that, then and then you can start phase 3. Or if you start here, you could save some time, but then these are missing, and FDA wants to see this timeline. Or you can do it here, but in parallel, you need to do this. Probably if you know that you are not going to be, you are going to be successful over here, what we are using in phase 3. Usually outcome of these 3 is usually in phase 3, the stability data and whatnot. But if you are confident based on your 2, 3, or 4 years of experience, you go it here and then do it in parallel. So what I'm trying to say is, if you have to save the time, you have to do activity in parallel. Taking risk, you might fail also. Okay, so that's the take-home message here. There, research is always about risk. You take risk, you expedite, but then if you fail, don't penalize your people, because you are taking the risk. You reward them instead of penalizing. And Novartis, we have the culture, we always reward. Even the molecule project fails, we have a system of rewarding, because they did their hard work. Molecule fails because of whatever reasons, toxicity or no efficacy, we cannot penalize it. And that's the culture we have at Novartis. And people here at Novartis, I can tell you that people are there from last 35 years. I'm there from last 15, 16 years. I left, but I came back because of the good culture. Culture is so good that you do your science so well independently, and nobody can stop you. And that's the culture we have. And very few companies, they do that, frankly speaking. And people usually, you know why people leave company. They don't leave company, they leave culture, they leave manager, right? They don't like people around, they don't like managers, and then they leave company. They don't leave company, they leave people they are working for. Always keep in mind, but that's a good lesson for managers. How can I do best so that people don't, oh, I'm leaving Novartis. No, you are not leaving Novartis. You are leaving that group, that culture, that manager for 2 reasons. One, culture is not good, or you are not being developed. You are not getting the development of, you are smart, intelligent, but neither you are getting promotion, or there is a limited growth. The only 2 reasons usually people leave. But you need to have a culture where, and the development path for the right people. You can't develop everybody. Everybody cannot become global head, or everybody cannot become head of R&D. And that's why people leave, which is perfectly fine sometimes. But majority of the time, within 1 year or 2 years, 3 years, somebody is leaving, means the culture is not good, or they are getting the better opportunity somewhere. Anyway, so in summary, selection of a project plays a key role, as you say. We have to define first in class, best in class, small market, big blockbuster, develop in-house, out-license. We do out-license. We are a big pharma. We do out-license too, because some asset we are trying to develop for multi-billion, and then we realize that this is for a small population, and we really cannot spend that much time. So we out-license. There are a lot of companies, they take it and develop it, because the whole point is to save and extend patient life. It doesn't matter who develop, right? And again, project management and scientific expertise play a key role. Okay, I don't go into more detail, but just wanted to highlight what I just said, the company culture, our culture, especially Novartis culture. Our CEO was, you might know him, few of him. World follows him. A lot of CEO follows him, because he's one of the unique. He's just 44-year-old, talented, super talented, unique, leader we got at Novartis, and he created and kind of creating unbossed, curious passion, a culture within the Novartis. We already had a great culture. He brought further, another angle to it, and we are all very honored to have him. And everyone within the Novartis is very proud of what we do, and for saving and extending patients' lives. So with that, I will take a pause. I think I might have gone further, but happy to take any question and answer. Thank you so much again.
Thank you, Sudhakar. Now the session is open for the question answer. If queries are there from the delegate side, as such, I would like to mention from my side that Dr. Sudhakar is a very humble and sincere person. As a friend, not only friend, he is like a brother to me, and we were a colleague, his roommate also in the Nasik College of Pharmacy. And it is a true example how a simple person or a simple boy from a small village can go to Novartis USA here and heading that institution. Okay, so that is the very important achievement what one can learn from him. uh, सुधाकर एक छोटे से स्मॉल प्लेस है शेमाल पिंपरी जो पुसद डिस्ट्रिक्ट है वहाँ पे. Okay, and his father is only teacher that time, and with great difficulty both of us could go and enter to the Nasik College of Pharmacy, and with great difficulties again, not from the academic point of view, but from the very few institutions was there that time. Admissions was very less. In Pune University, only 3 colleges was there. And after DPharm admissions, taking admissions into colleges like Karad, Nagpur, and College of Pharmacy, Nasik, the government aided institution was there. So we were very lucky. And with his hard work, he has shown that from such a small place, rural place, one can come to Nasik, Nasik to Mumbai, and Mumbai to abroad, USA, and working for such a nice place, okay, and in a company. And he's doing very hard work, sincere work, helping students from his old colleges also, as well as from his villages also. He's helping regularly. He's a very kind person. He has written one book also recently, very nice. So I thank Sudhakar, and for listening to him. One more special person, his friend, Dr. Vijay Shastri. I request Dr. Vijay Shastri to unmute himself and keep the video on. Yeah, Dr. Vijay Shastri, welcome. He is also one
Thank you so much, Sharad, Dr. Sharad. It's a great pleasure once again to hear Dr. Sudhakar, my close friend, as well as Dr. Sharad Wakode. We all were classmates at Nasik College of Pharmacy. Thank you so much for inviting me. Please continue.
Thank you. Dr. Shastri is a very good teacher of pharmacognosy, and he is an entrepreneur also. He has started his own pharmacy institution in Jalgaon. Thank you, Dr. Vijay.
Thank you so much.
Any delegate wanted to ask a few things?
Hello, Dr. Sudhakar.
Yes, sir.
Yeah, sir, this is Sandesh from Maliba Pharmacy College, Gujarat. Sir, what is your take about India on NCE?
Sure.
NCE, why Indian scientists doing very well outside, and a lot of credit and success goes to their honor, but in India, the NCE research is dying. So
yeah, no,
could you put
sure.
your words on that?
Absolutely, Sandesh sir. Thank you so much for a very good question. One of the most important thing is infrastructure. You know, one thing what we need for basic research, new chemical entities, having right infrastructure. You saw on one of my slide, right expertise first of all, which is possible. You know, India has a basic science a lot. You know, we have good biology. You know, we have good chemistry. So basic science is there. Now, infrastructure means, of course, instrument and the funds require investment. And you must have heard me, and if not, if you put 100 crore, there is no guarantee that you will have a success from that molecule. There is no guarantee. As you have seen, you put out of 100, only 10 molecule goes to commercialization. Now, if we are putting 100 crore, just an example, or 1,000 crore, and then you really do not know whether you will get a molecule out of it and where you can money. Who will invest in that, right? So one of the reason I would say, major reason I would say is return on investment. That's the only major reason. Science, I don't think so doubt, because as you said correctly, a lot of talented Indian colleagues are there abroad, those who are adding value, but not in India, because their talent is not being explored there. If I come back tomorrow to my home country and I say, let me do something, and they'll say, what are you talking? We don't have money. If I ask, oh, I need 500 crore to do XYZ, they'll say, oh, what? And when we will get molecule, I'm not sure. We may get or get not. And they'll say, are you sure? Who will give you? But in United States, it's different. It's in United States or Europe, they are rich people actually. First of all, very rich people. And out of that, even 10% thing I want to invest in pharma, it doesn't matter whether I get it or not. At least the benefit of rich investor people, if they don't get anything, they don't invest, they are tax deferred. Here you cannot do anything. Every dollar you earn, you have to pay taxes. Not like back home, So then at least you'll say, okay, in simple word, I'm investing money to save somebody's life, and if the money won't come out, at least I can pay for taxes, and it's okay because I have a lot of money anyway. That mindset is there. So in summary, I would say the return in investment, that is the major reason not to have new chemical entities from back home. I hope that helps.
Yeah, sir. Thank you very much, sir.
Welcome.
The seminar is open for question and answers. Anyone else has the question?
Yes. I have questions. Should I talk? Shall I talk?
Yeah, please unmute yourself as well as your video.
Yes. Myself, Dr. Satish Polshettiwar from MIT Pharmacy, Pune. It was really a very informative and thought-provoking session that you talked on various issues of drug development, drug discovery. So my question is that in terms of commercial viability, so in Indian industry are lacking with that, okay? Because just now we talked about that Indian industry are facing the problem of that investment and the risk and all. They don't want to take risk. Only Cipla could produce one drug, that is anti-diabetic drug. That is only one molecule in India. But I would like to ask one question to you: how this in silico modeling and CAD technology would be helpful to bring this drug discovery process very faster, so that it can be that industry can survive. So could you talk on?
Yeah, CAD. Thank you so much, sir, for asking the question, Satish.
Yes,
CAD plays an important role. You saw in one of the slide, that is one of the backbone, because we have thousands of target, you have thousands of molecule, and you really cannot go blindly making molecules. So CAD helps to define a different scaffold, narrow down from 10,000 to maybe 100 or 10 scaffold, and from 10 scaffold you put different functional group and go from there. So in nutshell, I don't have expertise in there, but that is one of the key to start off the discovery. Once it is done, then you are good. And without that, we cannot do discovery. So CAD in India is, although it is powerful, but unfortunately, students may not have that many opportunity, but they can go for postdoc, or they can go abroad, and then that might be happening. So I see the people who are doing in CAD, they have a lot of opportunity abroad, and they won't stay in US. That's for sure. In India.
Yes.
Makes sense.
So that could really make that drug. That means we can save the money and we can develop. Indian company can come forward and they can bring the molecule in market. We could see maybe we expecting the molecule in coming days from Indian company.
Absolutely, because that is one of the key parameter. And then later on, as you know, out of 10, only 10 successful.
So academic university will play a vital role in India. And now we have to focus on academics.
Absolutely. And then the research industries, they can collaborate with academia and get that knowledge, know-how, and that expand. So that link has to establish.
Yeah, that we have to just bridge that gap. That is a requirement. I would suggest professor to take forward this. Really, this is a really need of our at this moment for our students. Bridge that gap.
Absolutely.
Industry academy. Yeah.
Good point,
thank you,
Satish sir. Yes.
Thank you, sir, for your valuable suggestion.
Any other question? Sir, I have a question for you.
Please.
This is a general question.
Go ahead.
We like you have worked on say 200 new chemical entities.
Yep.
And over 10 diseases. Now we pour in so much of efforts, academic efforts, intellectual efforts, then economical, inputs, and we develop a chemical entity, and it survives only for say 5 to 10 years. There are very few drugs which survive beyond 10 to 15 years. So, isn't it that all these efforts are, making the humans as guinea pigs for 10, 15 years, and then after that, again a search for the new chemical entity, the same process is repeated under the pretext that we are developing something new now.
Yeah, no, I totally agree. Very good point. What happens is, this is all usually voluntary, right? As you said correctly, in drug research, there is no compulsion. Patient participation, volunteer participation is always voluntary. The way you have passion to teach student and bring a star, I have passion to discover medicine. Volunteers, they have a passion that, oh, I would like to be part of that clinical trial. If
Sir, sorry to interrupt you. I'm not talking about the volunteers. I'm talking about the general population. Say I have born in the era of 1950 to say 1980. I'm there, or 60 years, you can say 2010.
Yep.
So whatever medicines are discovered, or whatever the doctor or whatever the scientist feels correct at that moment is given to me. And I'm trying to alleviate from the pain or whatever disease. It may be successful, it may not be successful. And after that, the drug which was said to be effective in say 1990 becomes useless in 2000.
I got your point. Yeah. No, you are absolutely right. See, what happens is you bring a really good point. Sometime, for example, and then maybe now I understood your question. For blood pressure, if our Enalapril, which is invented in 1980, takes care of blood pressure, right? For example, why do you give the highest expensive current molecule, right? Totally agree. So what happens is, I'll tell you, many times, especially in, of course, developing country, doctor usually do exactly they practice like what your old new chemical, depending on the depending on the blood pressure and condition and situation. If old molecule has a potential to cover, and I'll give you a simple example. It cover that particular disease or condition. Why not just give that one? Let's not get complicated, But what happens in EU and US, all this, for example, blood pressure and all, they do practice like give me simple old molecule, but majority of the new chemical entity and new targets, they are like, what are the new diseases arriving or indications are there, they go after that. So there is no old, era will work. That's why this new target, that's why new company. But you are absolutely right. If anything can happen with a simple molecule, just treat that. Don't go into the complex one. I completely agree with you.
And not only that, see the cost, the cost is also increased. I feel that it is a nexus of the pharmaceutical companies to bring in new molecules all the time, and the molecule which they have worked on, they themselves declare that it is useless now. You work with this. So for those 10 years, that population is treated, and for the next 10 years, whatever is preached by the pharmaceutical industry to the doctors, that is prescribed, and then that is considered as good. Say for example, statins came into the picture, and these statins were prescribed rampantly, and then later on it came that the statins are no more required.
Totally agree. I think
So it's a sort of earning gimmick for say 20 years or so, and then you declare that this is useless, start with something else.
Exactly, and you could say a simple life example. Remember when I was in a college, that time there was no cell phone, but there was, I think, in 91, 92 cell phone came, like such a big cell phone we had, and we would carry. Now you have very tiny Apple iPhone, right? Nobody will touch that because you have really smart device right now. So this, I think, as you said, as a function of time, one is like a commercial. It's a simple example I gave of a phone. Like people really go for easy based, safe, easy based, safe to use, for example, iPhone, smartphone. In medicine, sometimes you have no choice, right? I mean, there are certain diseases, and like one of my friends' mom, she had a breast cancer, and she has to go every 2 weeks to clinic and take infusion for half an hour, and very painful because there is no other indication. It's a typical type of breast cancer. So I mean, the point is, like, there she has no choice only, and the medicine was invented by BMS like 1995. Not getting the name, but it's a really cocktail of different.
Is it Herceptin?
It's a parenteral. Is it parenteral? If it is parenteral, then it could be.
It's Herceptin probably.
Exactly. So there is no choice. Remember, this is old one, but somebody comes up with the new one, then of course that would be gone. So you bring really good point. We know sometime depending on the situation, it's a combination of saving patients' life. It's combination of earning money. It's combination of investor. It's combination of insurance company. So in the end of the day, it's kind of
or
as you
is
said
it
correctly.
survival of the pharmaceutical industry?
Exactly, ma'am. Exactly. It's everything. You could combine all those factor and bring. I mean, ultimately, what we want is, like, for example, in Corona situation. I mean, in Corona, US is paying 5 billion, 500 million, half a billion to all 5 different companies who are in phase 3. Like, they need 100 million doses first from each of them. They paid half a billion, 1 billion. Now that money is required, of course, for development of their work, as well as when the vaccine is available, ready to use. So you can imagine it's a matter of, who has the money and who pours money to get the. Now everybody needs it, but it's just a matter of time, right? So in nutshell, you bring a good point, but end of the day, what I want to say on this topic is, if you close eyes and somebody needs medicine and somebody is dying, it does not matter what it takes and how many players and what gimmicks and all that. What I want is, I want to save that patient or that's life. That's it. It doesn't matter whether it comes from. That's what I see. Rest of the thing, as you said, I usually don't get into that one. Invent it, cure patient, and move on. Whether 10 people take benefit or one, none of my business. I want to save that patient life. That's how I look at it.
Great.
Okay. All right. Thank you.
हमको अपना जूनियर मिल गया तो she is asking. Madam is also from Bombay College of Pharmacy.
Yes.
तो मैडम आज ज़्यादा इंटरेक्टिव हो गए हैं. Nice. चलो, it was good to interact with you. Thank you, Sudhakar. Madam, please.
A very lucid presentation in a simple, stable voice, step by step, keeping composure all the time. So it was a wonderful presentation with all the details and, good insights into the subject, because you are actually doing it. Someone not doing and talking about it and doing and talking about it is something different. So it was a wonderful presentation, and our delegates would agree that they have got a good depth of the knowledge in development of new chemical entities and clinical research further. It is our pleasant duty to honor you with a virtual bouquet of flowers.
Oh, so nice.
And, let me just kindly accept it. Oh,
Thank you so much, ma'am.
It's a certificate which has come. I'll just come with a bouquet as well. Let me just. Yeah, kindly accept the floral bouquet as well as the certificate.
So
we
nice
would
of
have,
you.
we would have loved to have you in our institute, but somehow this corona, we are thankful also because we cannot afford calling you from USA to Delhi to deliver the lecture for our participants. But because of the corona, we could take advantage of the online program and ask you to get up early and deliver for all of us. It's so nice that you have taken so much of pains for all of us. Thank you so much, Dr. Sudhakar.
Pleasure, no problem. And I would love to come and see. I have been telling Sharad that whenever I come to Mumbai, I will make a special trip to Delhi. It never happened. Usually I come every once in 2 years, and that to be for 1 or 2 weeks, very tight schedule.
Yeah,
but I will make it sure that I have to visit Sharad there, because we stayed
Next
together
time-
for 4 years.
Sir,
Definitely
next
I
time
will.
what you can do is, instead of taking a flight to Mumbai, take the flight to Delhi and then you will reach to Mumbai in any way. Opposite
Absolutely,
will
will
be,
do.
difficult. But if you
Yes,
take
yes.
the flight to Delhi, it is possible.
We will love to see all of you. Okay?
Yeah. Even we will love to host you.
Thank you so much, ma'am.
Okay.
Great to be here.
Yeah.
Thank you.
There is a announcement for the delegates. We are ending with the third day successfully with wonderful presentations by 4 speakers covering wide variety of the topics. Morning we started with the designing of the drug and how the softwares are used. Later on we went to the impurity profiling, and the day ended with how these new chemical entities which are developed, then how they are converted and by QBD taking care of the impurities, and then later on how they reach to the formulation stage and then to the patient and then to the targeted cell of the affected area. So it was a wonderful journey from the morning till evening. And tomorrow, all of you should log in at 9:15 because we have a speaker again from USA, and she would be delivering at night 12. So we should not delay her beyond night 12 because it becomes very tough for them also. So we have an early session. IT team, kindly take note that we have to log in at 9:15, and delegates, you also have to take a note that you all join, have early breakfast, and then join for the lecture. Thank you so much. We declare that we are ending the third day's session. Good evening to all of you. Have a nice evening.
Thank you, ma'am. Thank you all of you for joining.
Thank you, sir.
Thanks, Sharad.
Thank you so much.
Thank you to all.
Thank you. Bye.
Hello.
Sandesh, you wanted to talk something?
Yeah, I just wanted to tell Sudhakar sir that I am also an alumni of CPN.
Oh, good. I'll convey him. I think he has left the meeting.
Left.
Thank you.
Okay. Thank you.
Yes, sir. Great. Thank you. Thank you, madam. I think